Or I've already introduced myself. I'm Jason Roberts. The purpose of this section is to just talk about, and if people aren't interested in coming back in, that's okay. My section is just to talk about the different types of therapies that are going to be discussed. So the things that I'm going to be talking about, so RNA knockdown, we're not discussing that today. In the sessions, but it is a therapy that will be emerging in time. And then the other two topics that we're going to look at, so gene transfer, that is the main gene-based technology that's going to be discussed in the sessions. And then there's also a session later this afternoon on gene editing when Intellia Therapeutics will present. All right, so RNA knockdown. There's two different types to knock down mRNA. So the two different modalities are antisense oligonucleotides and small interfering RNA, or siRNA. In terms of how these work, so these are prime time now in clinical medicine. There are drugs that people use now with these modalities. So siRNA and ASO, the way that they work is essentially they're ways to just knock down the mRNA before the mRNA is able to be translated by the ribosome. So ultimately, the protein is not expressed. So I won't go through the kind of nuanced details that's different between siRNA and ASO, except to say that these molecules bind with the mRNA. And I have to say, it's unfortunate that people aren't. Anyways, okay. So the mRNA isn't ultimately able to be expressed. So what diseases are potentially amenable to RNA knockdown? Ideally, there are potentially ideal targets are pathogenic gain of function variants and dominant negative alleles. So this is where. And if you're using this approach, you have to be losing a portion of gene expression must also be tolerated. So this isn't going to be good for a lot of inherited arrhythmia and cardiomyopathy conditions where, so for gene transfer, you're treating these conditions because they've lost part of their the gene expression. Here you're so long QT syndrome, the major subtypes, it's not going to work for ARVC, ACM, or PKP2, DSP, LMNA. Those things aren't going to work. But where they may be potentially helpful are things like phospholamban cardiomyopathy, where you seem to be able to lose about 50% of phospholamban and potentially TMM43 as well. They're particularly ideal when genes not needed at all. So where these have emerged first are for lipids. PCSK9 is a gene that is important in cholesterol metabolism. And it doesn't seem like adults need any PCSK9. And in that context, there's an siRNA that is now being used clinically called inclisiran that is given once every 6 months and knocks down PCSK9, both alleles. It's targeted to bring down both. Another area is in lipids is Lp(a). So this also predisposes to atherosclerosis. It doesn't seem like you need Lp(a) either. So this was an ASO from Ionis Pharmaceuticals that now Novartis owns it, but works really well at reducing Lp(a) expression, and this is more recent data with siRNA. So is this cardiac prime time yet? Not yet. Delivering these molecules, they're able to get it to the liver very well. They're working on delivering it to the heart. It depends on how you tag these molecules in terms of where they go. So there's promising preclinical data. So this was a phospholamban mouse model where they developed, so as people mentioned, I think already, so phospholamban, there's an R14del variant that accounts for a large proportion of phospholamban cardiomyopathy. So they developed an ASO specifically for R14del. Knock that down, you have 50% of normal phospholamban expression from the normal allele, and the mice seem to be completely protected after treatment for that. So, and I think the same potential thing could happen with TMEM potentially, but they're still working on getting those to the heart, but it's coming. So gene therapy, so this is the main thing that I wanted to focus on today, because these sessions that we're going to have, most of them are on gene therapy or gene transfer. So the nomenclature it varies a lot, but I view gene therapy as gene transfer. You're delivering a gene to an organ with a viral vector. So this is a general kind of overview of it, and I'll highlight in just a minute. A key thing with gene therapy is the delivery vehicle. Are you able to get the gene effectively into the cell, and is it ultimately safe? So the vectors that are going to be discussed today are all AAV, adeno-associated virus. They're taken up by the cell, then into the nucleus, and then generally established as an episome, so they're not integrated into the genome, and then the wild-type protein or the normal protein is expressed, and if a disease has developed because you're missing one copy of the gene, this is a way to potentially cure the condition. So yeah, I kind of went through this. So delivering a gene with a virus into a cell is called transduction. So transformation is getting DNA material on its own into a bacterial cell. Transfection is DNA material into a eukaryotic cell without a virus. Using a virus, it's called transduction. And it seems to work overall pretty well. There's limitations, so size limitations using AAV. The gene can't be too big. So Titin cardiomyopathy is obviously the major cause of DCM, but Titin's too large to get in an AAV vector. And I went through in terms of what happened. So I mentioned in terms of what diseases are amenable to gene therapy. So you've lost one normal copy of the gene, and you're replacing it. What are not amenable to gene therapy? So gain of function variants and dominant negative as well. So importance of the delivery tool. So we'll hear a lot, I think, from our industry guests here today about their tools. This is important. So gene therapy back in the 90s, everyone was really excited about it, but it had to be shut down. Back then they were using adenovirus, which is different than adeno-associated virus, and there was deaths because of dramatic immune responses to these viral therapies. So fortunately, things have come a long way over the past 25 years. The ideal properties of a delivery vehicle, so low immunogenicity and efficient organ-specific delivery. So AAV does not integrate into the genome. There's lentiviruses, which are not going to be discussed today, that are mainly used in the setting of hematology, do integrate into the genome. If they integrate into an appropriate part of the genome or potentially have a very active promoter, there's the potential for induced cancer. But fortunately, AAV does not go into the genome, and that hopefully that we won't see those kind of things coming up. So we can kind of, so AAV, not adenovirus, is what we're going to be hearing about today. And yeah, it's not known to cause human disease. And after it gets into the cell, as I showed in the diagram, it gets established as an episome, not integrated into the genome, and then it expresses the protein. So in terms of considerations for AAV-based gene therapy, and this is just kind of a broad talk, so the companies don't need to go through things. So patients can't have AAV antibodies. So there's a portion of patients that have AAV antibodies. They're not eligible for these therapies. And you can only get AAV therapy once. So after you've had it once, you'll have antibodies, and then would subsequently mount an immune response. So yeah, there's been lots of excitement in this area. So we're going to hear a lot about, I think, PKP2 ARVC today. So this is from some of the companies as well, in addition to some academic groups, talking about how PKP2 gene transfer can potentially cure ARVC. We're going to hear about Danon disease, getting LAMP2 into cells can potentially fix this condition. And this is what's farthest along. So Rocket will be showing human data for their Danon gene therapy program. And this is just in terms of arrhythmia, the preclinical stuff that is not related to the companies, although it's somewhat related to Solid's presentation later. So this was just in the setting of CPVT. 10 years ago, Sylvia Pereira was showing us that if you get Cask2 into Cask2 CPVT, it can cure them. I won't, just in the interest of time. So Mike Ackerman, who we all know, has a very, very creative way to try to treat a lot of conditions with a type of gene therapy. One of the challenges with these conditions is that you have many, many different variants potentially for DNA editing. How do you address all the variants? He has this approach called suppression replacement, where you deliver DNA such that there's a small hairpin molecule that will knock down all of the endogenous genes. So if it's long QT1, knock down all of the endogenous KCNQ1, so both the disease allele and the wild type allele that's endogenous. And then the delivery vehicle also contains a normal KCNQ1 that will completely replace the patient's KCNQ1. So that could potentially be used for a lot of different conditions. So that's AAV. So that's the main gene therapies. That's the main thing that we're going to be hearing about in subsequent sessions. And just in terms of brief prep for the Intellia session, so CRISPR. So I think everybody's hearing about that now. So this is here in humans now. It's being done, and the first CRISPR-based therapy was just recently approved for sickle cell in the United States. So how does CRISPR work? There's a few different versions of it. So what Intellia will be talking about is version 1.0. It's a double-stranded DNA break that essentially destroys the gene effectively or eliminates expression. You get a double-stranded DNA break, something called non-homologous end joining. The cell essentially panics, puts it back together, and usually results in a frame shift variant that ends up resulting in nonsense-mediated decay and lack of, and you don't get the protein expressed. So I'm not going to talk about their programs. I'll let them do it, but their programs are essentially knocking down a gene with CRISPR. There's newer types, base editing. It's like a pencil eraser. You can change a C to a T and an A to a G. And then prime editing is the one that has the potential, most potentially, but it's the one that's earliest along. It's like a word processor. You can put anything in that you want and take things out as well. And this also, there's a version, an adaptation that involves an integrase that enables you to get a whole gene potentially into the genome in an effective way. And then there's epigenetic editing. I won't go into that. So I think we'll just finish with that. CRISPR, they get the material in with something called a lipid nanoparticle in general, not with an AAV. So anyways, that's just, we felt it was just important to just provide an overview of things that are going to be talked about today, just that each company doesn't feel the need to run through it. So the main area is gene transfer, so generally delivery of a wild type copy of the gene, and helpful for correcting pathogenic haploinsufficiency. So I think I'll leave it at there, and then hand it over to the moderators and speakers for the next session. Sound okay? Great.
All right, good morning, everyone. It's my pleasure to moderate the session alongside Dr. Atala, who is online. Unfortunately, he had clinical duties to attend to, so he'll probably join us for some questions at the end. And we'll also have a chance, I think, after all three speakers to be able to go through some questions people have. And also Paul's here, who's going to be moderating as well. So the first speaker that I have the distinct privilege of introducing is Dr. Jeff Haroldson. He's the Executive Director of Medical Affairs for Tenea Therapeutics. Jeff has over 20 years of experience in the biotech industry, with the majority of his focus on rare diseases to help bring precision medicines to patients to address a significant unmet need for the currently available therapeutic options of these individuals and their families. So, Dr. Haroldson, we're happy to have you, and I think it's wonderful that we finally get to talk in this area of medicine about more than just beta blockers and ICDs, because that's been quite inadequate for many people for many, many years. So I think it's super exciting to be able to talk about potentially curing these diseases before they even have their phenotypic onset. So thanks.
Great, thank you so much. And it's an honor to be here. Thank you for inviting me. We're happy to be sponsoring this meeting as well. And on behalf of all my colleagues at Tenea Therapeutics, who work very hard every day to get to where we are. So a really exciting time with gene therapies for cardiomyopathies. So I assume most of you, I know some of you know Tenaya Therapeutics, but I imagine most of you don't. So I'm going to do a brief introduction of who we are, what we're working on, and I'm really going to focus on our 2 lead gene therapy programs, and that's TN-201, which is our gene therapy for MYBPC3 associated HCM, and then our TN-401 gene therapy program for PKP2 associated ARVC. So who are we? I mean, we really are grounded in patients first. It's our #1 core value at the organization. We have a phenomenal patient advocacy team who actually brings patients and their families into the organization frequently and through webinars. So even for those who don't have a healthcare background in the organization, they've really learned what the burden of disease is like, what it's like to live with the disease, and what the hope is in terms of unmet need and therapeutics. So it really drives our purpose in terms of developing molecules, gene therapies for rare diseases, and trying to bring potentially curative therapies to these patients and families. So I'm going to introduce the company. There's a lot going on here, but I'm just going to emphasize a couple of things. #1, Tenaya Therapeutics is singularly focused on the heart. So in all of our preclinical programs, including our 3 clinical stage programs, it's all cardiovascular disease, mostly focused on rare cardiovascular disease with a genetic cause of the disease, but we do also target some more prevalent cardiovascular diseases. As such, our leadership, we have several cardiologists who work at Tenaya. We also have gene therapy experts, experts who have been worked in rare disease for years, and we really feel like that's a really terrific union and collaboration internally to really drive this forward. We have on the bottom left, or actually that just popped up, 100+ employees. We're a small but mighty company, a little over 100 employees. Tenaya is based out of South San Francisco. And also on the bottom right of this slide, I think, which is important in developing genetic medicines, we invested in developing our own manufacturing facility to really have control over product purity, safety, quality, and that's really essential, we believe, in terms of delivering safe and effective gene therapies for patients. Not sure how intimately aware of everyone in the room is with gene therapy, but I'm going to focus just on AAV gene therapy. It's really relatively new in the cardiac arena. Our HCM program is out ahead of our PKP2 program, and that has been a pretty big learning that I've discovered in talking with HCM experts. And so AAV went from being discovered in a lab in the mid-1960s to now well over 20 years of experience. You can see that over 5,000 patients have been dosed worldwide. The majority of those have actually been in a rare neuromuscular disease with a product called Zolgensma for spinal muscular atrophy. At least in the US, we have 8 FDA-approved products now, gene therapy, AAV-driven gene therapy products, and really good follow-up, more than 15 years of follow-up in certain disease areas such as hemophilia. When you look at clinicaltrials.gov across the globe, there are over 200 AAV gene therapy trials underway across the globe. You can see the majority are in the US and elsewhere. And on the bottom right, just to put it in perspective, about a dozen of those now are focused in cardiovascular disease and cardiomyopathies, and you're going to hear about a few of those today. So just to dig in to our very first program, it's the TN-201 program. We're calling the trial My Peak. So Tenaya is a mountain range in the Yosemite area in California. So we have very cute titles for our trials, all related to climbing mountains, peaks, and ridges. So we have, so our first TN-201 is targeting specifically individuals with HCM with mutations in the MYBPC3 gene. So we know that in HCM, roughly almost 60% of all cases of HCM are driven by genetic mutations, and of those, the MYBPC3 gene is the most common. ClinVar, actually in a recent report, put out that out of all HCM variants, almost half of the pathogenic and likely pathogenic variants in HCM are in the MYBPC3 gene. So that gene encodes cardiac myosin-binding protein C, which is a really integral component in the thick filament primarily, that thinking of it simply as like a braking mechanism. So when that protein content is low, And we know from surgical myectomies in adults with HCM, studying protein content, that protein content is about 60% of normal. So it is reduced. You see the phenotype as a result of that. And without that protein on board, that you see the hypercontractility, hypertrophy, relaxation time delayed, diastolic dysfunction, all the sorts of things that we see in HCM. So we really believe that bringing a transgene to the cardiomyocytes to help restore near normal levels of protein should restore cardiac function. Jason already went over this. I would say my illustration maybe is a little more interesting probably with colors, but the idea here is you take the AAV, we use AAV9 serotype. There are several AAV serotypes. Different serotypes have different tropisms in terms of targeting different tissue. AAV9 is very well established in targeting cardiac tissue. Essentially, the viral DNA is removed, and then a full copy sequence of the transgene of interest, in this case MYBPC3, is inserted, along with other cargo, regulatory elements, the promoter to help the gene initiate transcription, and so forth. This is delivered intravenously over a couple of hours infusion. It's a one-time administration, as Jason mentioned. And again, the AAV9 vector is really, really effective at targeting cardiomyocytes, undergoes uncoating, and again, forms episomal DNA, does not integrate, and restores cardiomyosin-binding protein and leads to restoration of function. The TN401 program, we also use AAV9, so I just want to pause on this slide and just at least give you the rationale in terms of why did we select AAV9. Number one, it's very well established in the market as far as efficacy and safety data. Most of that, again, is in young children with SMA, but also Duchenne muscular dystrophy has used the AAV9 vector quite substantially now, too. Novartis has just recently reported up to 8 years follow-up now in these SMA patients. So we really know what you're getting into as far as efficacy, know what safety signals to look for. Currently, it's only capsid with validation in human hearts with patients receiving gene therapy with the ability to transduce cardiomyocytes and leading to durable gene expression. And we'll hear from Dr. Schwartz, I'm sure, on some of that data with the Danon disease. And then also, it's the most extensive body of clinical evidence, preclinical evidence, and independent studies in the PKP2 disease model. And we just recently, at the ASGCT conference in May, published and put together some of our own data where we actually really wanted to dive into this a little deeper, looking at AAV9 versus other capsids as far as transduction and expression. And we found that AAV9 leads to higher expression. And we really feel that that's important because with AAVs, even though they are less immunogenic than adenovirus, we still see these complications and potential risks. So with higher expression, we're able to come in at lower doses, and we certainly hope that that translates into a better safety profile for patients. One slide on the T01 preclinical data that helps inform and shape the dose going into the clinical trial. These are some key efficacy readouts that we looked at after we looked at the 3e to the 13, so 3 times 10 to the vector genomes per kilogram. Much different way of thinking in terms of how you deliver these therapies with a dose in the MYBPC3 knockout mouse model. And you can see from moving from left to right, the black is the wild type, blue is the control animal not treated, and then the 3 orange bars. We had different doses, and that middle orange bar is our 3 to the 13, so where we saw near maximal efficacy in terms of reducing LV mass, and the middle bar, those animals actually saw an increasing ejection fraction and then stabilized over the duration of the trial. The study in the blue bar, you can see that the ejection fraction continued to rapidly decline. A lot of these benefits led to 100% survival in these animals treated with TN201. It's overlaid on top of the wild type, or the lighter color across the top on the right. 100% survival to the point of sacrificing the animals at 18 months, whereas you see all the animals, the control animals who were not treated, did die shortly after a year. So brings us now to our trial, the MyPeak phase 1b trial. We have initiated dosing in patients, and and the first patient was dosed in the fall of last year. This is, as 1B in gene therapy trials, this is primarily a safety trial. We're looking at safety tolerability. This is still the dose finding with the opportunity to, with the data safety monitoring board, based on findings, efficacy, safety signals, we may move into a slightly higher dose, so that's to be determined. Right now, we are starting this trial in adults with non-obstructive HCM, got to be symptomatic, and an ICD present at this point, and EF's higher than 50%. The neutralizing antibody titers are important. That's part of the eligibility criteria. This is an open label design, so all patients entering this trial will receive the gene therapy. There's no placebo control at this point. And you can see the schematic in terms of looking at 2 different dosing cohorts with the option to continue dose expansion up to a total of maybe around 15 patients through the phase 1B cohort. Some of the outcomes that we're looking at are endpoints, again, safety and tolerability. A lot of the usual suspects, for those of you who are familiar with the HCM trials with the cardiac myosin inhibitors, looking at all the structural and hemodynamic changes, exercise capacity, KCCQ score. But of course, the one thing that's different here is we want to make sure we'll be doing biopsies in these patients to ensure transduction and expression. It's a 52-week study. But then it's followed up with a 4-year follow-up with much less frequent assessments. Here's where we are currently across the United States with clinical trial sites. We are not currently in Canada, but we have several leading cardiomyopathy centers involved in the U.S., and even despite that, we know that trials like this are going to demand travel within or even outside of the United States. So our clinical development and clinical operation teams are fully prepared for that sort of situation. So jumping to our second program, that's in the TN-401 program. This trial is called RIDGE 1, and this is looking specifically at the PKP2 population, where you all know almost 40% of all cases is driven by a mutation in PKP2. A significant unmet need. You know this disease. A lot of these patients present with sudden cardiac death and a lot of anxiety around ICD therapies and so forth. So again, I think a really important patient population where there's a significant unmet need. In our preclinical models, again, a dose ranging study. I think I have some things to fill in here. Yes. So just looking at, again, some key efficacy parameters, we are also bringing in the same starting dose at this level based on preclinical studies, moving from, again, left to right, that 3E13, again, near maximal efficacy in terms of reducing arrhythmic burden, improving ejection fraction in the mouse model, and then also reducing RV enlargement. The AAV9 dose that we found with this near maximal efficacy is where we achieved transduction at about 1 vector genomes per diploid genome. So the biopsy, the tissue in these animals, of course, it's all mushed together. So you're getting different cardiac tissue, roughly half are cardiomyocytes, half are non-cardiomyocytes. But on average, this roughly equates to about 1 copy per cardiomyocyte. And so similarly to the TN-201 program, again, the IND was cleared at the early part of this year. So we are now moving into that phase where we will be dosing our very first patient in the second half of this year. So very exciting time again for gene therapies in the PKP2 population. Again, safety and efficacy, we're looking at adults again to start out this trial, symptomatic adults, looking at least a baseline PVC count of 500, keeping the EFs above 50% at this point, and NYH classes between 1 and 3. And again, open label. So all patients entering the trial will receive the gene therapy. So looking at a lot of safety signals, changes in PVCs, non-sustained VTAC, frequency of shocks, VTs, all sorts of biomarkers, and patient-reported outcomes. We have a few different quality of life assessments that we're using in this trial as well. Not as many active states. Again, we are not in Canada, so my apologies, but we could talk to Jason and Dr. Crown about this. But again, so this is where we are, and we're committed also to bringing this program to Europe and probably initiating in the United Kingdom as well. So, but this is where we are now. We've done a lot of work here to get where we are, so we're super excited about this, and hopefully you guys are too. Got my email there. If there are patients in the audience, we have an excellent patient advocacy team to reach out if you have questions. And we also have a couple of websites to learn more, arvcstudies.com, hcmstudies.com. So pretty easy to remember, and that's it for me. Thank you.
At this point, I'd like to invite Dr. Jonathan Schwartz on behalf of Rocket Pharmaceuticals. Dr. Schwartz has spent the past 25 years developing scientifically based targeted therapies to more safely and effectively address deadly diseases. He's a founding officer of Rocket Pharma, where he has advanced gene therapies for hematologic and cardiovascular disorders during the past 8 years. Welcome.
Thank you. Good morning. It's a pleasure to be here. Canada, people are friendly. Coffee is bitter, but that's better than the reverse. So over the next few minutes, I'm going to spend a bit of time discussing the Rocket Pharma sponsored gene therapy programs in Danon disease, PKP2 ARVC, and time permitting, a little bit about BAG3 dilated cardiomyopathy. Let's start with Danon disease, which is an X-linked disorder that results in impaired autophagy. Mutations in LAMP2 prevent adequate lysosomal autophagosomal fusion, which is essential for autophagy, which is the way cells digest and get rid of unwanted material, senescent proteins, damaged mitochondria, etc. And when autophagy is deficient, all that material builds up in autophagic vacuoles, which create a lot of disarray in the myocardium. And this results in a very profound and aggressive cardiomyopathy. Specifically in males, this usually results in a very extremely hypertrophic and rapidly progressive cardiomyopathy with median survival around age 19 or 20. Median survival for females is approximately 2 decades longer, but there's a great deal of variability. There are also extracardiac manifestations such as skeletal myopathy, although those tend to be less severe and not generally life-threatening. Our phase 1 study was conducted over recent years and involved RP-A501, which is an AAV9 containing full-length LAMP2B gene. Patients were eligible if they were male. We started with those who were at the highest risk for progression and adverse outcomes. Patients were excluded if they had high titers of neutralizing antibodies against AAV9 prior transplant. And during the study, prior to the pediatric cohort, we also implemented an eligibility criteria requiring preserved left ventricular ejection fraction. More on that momentarily. We treated 3 cohorts of patients, starting with the cohort 1, which is the low-dose adult cohort. That's a bit of a misnomer because that cohort was age 15 and above, and even though it's a low dose of 6.7 times 10 to the 13 genome copies per kilogram, that's still a lot of vector that's being infused into human beings. So it's low only relative to the other dose. That was the first cohort treated, and therefore we have the longest follow-up out to 36 months as of about a year ago. We also treated 2 patients in the higher dose adult cohort. That's not a dose that we're continuing. And we have follow-up now out to 24 months in that cohort. And then 2 pediatric patients who were both around age 12, who also received that 6.7 times 10 to the 13 dose, with follow-up now out to 1 year or more. Very importantly, of these 7 patients, 6 are followed up long-term and have derived benefit. The one patient, patient 1007 in the adult high-dose cohort, had more advanced disease and had progressive cardiomyopathy, required a transplant after about 6 months. So we'll present safety from all of the patients and efficacy from the 6 of the 7 that did derive benefit. This is a table that illustrates some of the baseline characteristics of the patients. As you can appreciate, the adult cohort patients were all between age 17 and 21 at study entry, and the 2 pediatric cohort patients in the bottom rows were around age 12. All patients were symptomatic with NYHA class 2 heart failure, and all patients had preserved left ventricular ejection fraction, except for patient 1007, who had diminished LVEF, likely indicative of more extensive fibrosis, which is why we believe that patient was not able to derive benefit from the therapy. As illustrated in the red boxes over in the right portions of the slide, these patients had extremely hypertrophic hearts at baseline, with just for the non-cardiologists in the audience, the typical upper limit of normal for LV mass is around 225 grams. Everybody in this study had more than that, including the kids who were part of the trial. Several of the patients had very high BNP at baseline. All of the patients had elevations in troponin, with the lowest elevation in high sensitivity troponin I being 7 times the upper limit of normal, indicative of the cardiac injury that's ongoing in this disorder. And we'll talk a little bit about the safety before going into efficacy. And this graphic illustrates the evolution of the immunomodulatory regimen that we utilize to try to prevent problematic immune responses in the days and weeks after vector administration. So in the first low-dose adult cohort, we started off just using corticosteroids at a relatively high dose, 1 mg per kg of prednisone, and then gave some sporadic tacrolimus based on hepatic enzyme values. In the second high-dose adult cohort with the 2 patients, we gave the same steroid dose but also used tacrolimus preemptively, as well as 2 doses of rituximab. Importantly, within those 2 adult cohorts, we did have 3 patients that had a steroid exacerbation of the underlying Danon disease skeletal myopathy, resulting in grade 3 muscle weakness. And fortunately, that resolved with a taper of steroids and supportive care, and fully resolved as these patients continued to be followed over the long term, but was something we sought to avoid going forward. We also had 1 patient in the high-dose adult cohort who had a grade 4 complement-mediated thrombotic microangiopathy, which resulted in high-grade renal failure and thrombocytopenia. Fortunately, those resolved as well over the course of a couple of weeks and completely resolved. But again, that's the sort of complication that we seek to avoid going forward. So for the pediatric cohort, we adopted the immunomodulatory regimen of a lower dose of corticosteroids, about half of the dose in the prior cohorts, 0.5 mg per kg, with an earlier taper starting around day 10. Sirolimus instead of tacrolimus, and rituximab. And the sirolimus and rituximab are intended to provide more synergistic B-cell inhibition to prevent antibody-antigen complexes, which trigger the classical complement pathway. And as a result of that, we did not have any substantial myopathy in the pediatric cohort. And we also didn't have evidence, didn't see evidence of substantive complement activation. Platelet count, which tends to be the very best bellwether for complement-mediated TMA, where those platelets remained in the normal range. Very importantly, with this regimen, beyond the 2 patients that were treated in our Danon pediatric study, we published an analysis of 15 patients receiving this same sirolimus rituximab lower dose steroid regimen. This included patients at University of Florida and NIH with GM1 gangliosidosis and SMA1. This was in the January Journal of Clinical Investigation. And one can appreciate across whether you're looking at the immunoglobulin graphs at the top, platelet count and D-dimer in the middle, or some of the complement moieties, including activated terminal complement C5b9 complex on the lower right, that the complement activation in the red group, getting rituximab and sirolimus, was markedly lower than in patients who were just getting corticosteroids. And that's the regimen that we'll carry forward in the phase 2 study, as well as in the ARVC trial. So we'll turn to efficacy now, and this is a representative cardiac biopsy of one of the pediatric patients. In the top row, you can appreciate that in the months and not represented years after therapy, there is expression of the LAMP2 protein fairly diffusely in the myocardium, and that expression is accompanied by a reduction in the autophagic vacuoles, those empty-looking holes that you can see in the middle row on the H&E slides or on the lower rows in the electron microscopy. And that reduction in these vacuoles is quantifiable, and I think you can appreciate, especially on the month 6 images in the middle and lower right, that a lot of the myofibrillar architecture appears much less disarrayed, much more normalized relative to where these patients were at baseline. In addition to seeing improvements in the myocardium, we saw improvements across a broad range of clinical parameters. So the troponins, which were all markedly elevated, were reduced quite remarkably. The BNP, which was at times elevated, reduced in those situations. And for patients who were normal or near normal at baseline, they remained relatively stable. The lighter green indicates stability. The darker green indicates improvement. Red indicates worsening. There's no red on the slide. The parameters of cardiac hypertrophy, such as left ventricular mass or maximum wall thickness, also reduced quite remarkably. Patients reported far fewer symptoms. The majority of patients at most recent follow-up were asymptomatic NYHA class 1 with fairly demonstrable improvements on the Kansas City Quality of Life score. A KCCQ improvement of about 5 points is considered clinically meaningful. We were able to demonstrate that in several patients, and improvements of 15 or 20 points in several others. This is just a graphic illustration, a little easier to read, of several of the patients with respect to left ventricular mass index, where you can appreciate that LV mass continues to to normalize in the months and years after therapy. That's something that we'll continue to track out to 5 or so years post-therapy. This is a graph also of the BNP. You can appreciate the phase 1 trial results for BNP on the left panel, where the patients who had marked elevations for the most part normalized, patients who were normal range remained there. And that's in contrast to what we saw on a prospective natural history study, where you can appreciate over anywhere from 3 to 30 months in Danon male patients of a similar age, these BNP levels continue to typically increase, in some cases very dramatically. So we were able to show the favorable safety profile for the low dose 6.7 with an appropriate immunomodulatory regimen. There were improvement across multiple parameters in 6 of the appropriately selected patients. By and large, continued signs of improvement or stabilization with ongoing follow-up, and we hope to provide further data updates for this phase 1 study potentially later in this year. And this resulted in our in agreement with FDA for a single-arm pivotal phase 2 study that is underway currently at centers in the United States and soon to be in Europe. This will enroll 12 patients, single-arm open-label trial using the 6.7 10E13 dose. The primary endpoints of this study will be assessed at 1 year and include LAMP2 protein expression in the myocardium, as well as an at least 10% reduction in left ventricular mass index. There are obviously multiple secondary endpoints that we'll continue to track, including quality of life. So moving to a topic that's most highly germane to this audience is PKP2 ACM or ARVC. And I really don't have anything to teach anyone in this room about this disorder. But we have come to recognize that this is obviously a disorder that affects people often in young adulthood. And despite a lot of advances in the field that were discussed and really very nicely elucidated this morning, we're still talking about a significant health burden, significant mortality risk, and particular subgroups of individuals based on ARVC risk calculations who may have a very high probability of ICD shock or other sudden death events over the course of 1 or 2 years, 3 or 5 years, and obviously a lot of things that potentially could be improved upon. RP-A601 is an AAV RH74, which is a variant that's very closely related to RH10. It looks like we have a MAC PC incompatibility issue with some of the graphics on this slide. And it's my fault for not QCing that early this morning. But at any rate, what I'll emphasize is that we did develop a number of animal models with an induced PKP2 knockout in mice, and we were able to show really tremendous survival advantage, improvement in left ventricular ejection fraction, improvement in right ventricular dilation at clinically applicable doses, and very importantly, This was done in mouse models in which the investigational vector was given after the induction of the knockout, in some cases 7 or 14 days afterwards, and which benefit was shown despite that. And very importantly, we were able to markedly diminish the incidence of isoproterenol-induced arrhythmias as demonstrated in the lower right, where you can see a lot of ventricular ectopy in the untreated mice and a nice sinus rhythm in the treated mouse cohort. The phase 1 study for RPA601 is underway currently, enrolling at UCSD and University of Pennsylvania. This is encoding a full wild-type PKP2A gene. We evaluated multiple AAV serotypes in the preclinical studies, and we went forward with the AAV RH74 construct that is a RPA601 because it had the most favorable safety and efficacy in relevant disease models across a range of doses. And really seemed like the most appropriate one to carry forward specifically for this disorder. AAVRH74 has been associated with a favorable safety profile at high doses in Duchenne muscular dystrophy, as evidenced by the experience with the Sarepta approved gene therapy product. We felt that it really was the logical choice. The study initial dose is 8 times 10 to the 13 genome copies per kilogram, which is a dose that we believe, based on the animal models, really confers a reasonable probability of clinical improvement. We continue to utilize the immunomodulatory regimen that I described, including rituximab, sirolimus, and lower dose steroids, to minimize potential for complement activation and other immune-mediated effects. For patients who are enrolled, this is daily monitoring in the hospital for the first 1 to 2 weeks with subsequent outpatient follow-up. The last program that we hope to bring to clinic over the coming year is in BAG3 dilated cardiomyopathy. BAG3 is a really complex and diverse protein that influences a large range of myocardial functions, from contractility to apoptosis to protein quality control. This is another disorder that frequently onsets in young adulthood, although sometimes even in adolescence or childhood. There's, for patients who progress to dilated cardiomyopathy, there's a 25 to 30% 5-year event rate with significant mortality over 3 to 5 years. Hopefully more on that program as we get closer to clinic. Preclinical studies are underway. A tiny bit about Rocket Pharma. The 4 clinical programs that we have presented data for, all of them are really ushering in a new era of genetic medicine. Two programs will potentially be approved this year for use in the United States, and that's in leukocyte adhesion deficiency 1, which is a devastating early childhood immunodeficiency, and Fanconi anemia, which is a bone marrow failure syndrome. 2 other programs are now in registrational trials with FDA and EMA alignment, and that includes red cell pyruvate kinase deficiency and Danon disease. Hopefully PKP2 will be next. The Danon disease results really represent the first gene therapy for a highly aggressive cardiomyopathy. We have multiple patient testimonials regarding gene therapy's effect on their lives. One of them is illustrated on the middle right portion of the slide. And there are many others that are available on our website or elsewhere, and the same is true also for the other programs in the hematologic disorders. I think we found that our work in the immunology and hematology gene therapy has really informed a lot of the cardiovascular gene therapy efforts. It's really an additive process. Rocket has, I think, a track record of dedication to patient safety. We collaborate with investigators around the world to try to continue to optimize the safety regimens that are utilized, and we are relentless advocates for research and support in rare and high unmet need disorders. On Rare Disease Day, February 28th or 29th, buildings around the world light up in rare disease colors. It started with our corporate headquarters at Empire State, and now it's everywhere, the Colosseum in Rome, and all sorts of other structures. This is, it matters to us. Obviously, all of this happens because of a tremendous amount of collaboration with colleagues at institutions all over the world. Several of them, those institutions and many of those colleagues are illustrated here, including Barry Greenberg and Joe Rossano, who've been really critical partners, and Barry Byrne at University of Florida, many people on the Rocket team, and of course, the patients and families who've been really tremendous partners and advocates in good times and bad. So thank you for your attention.
Great. Thanks so much to both of you for the incredible presentations on your current projects. I think we probably, according to Jason, only have just a couple minutes for questions because we're running impressively behind. But maybe just if the speakers can keep their responses to maybe 30 seconds, we'll have time for 2 or 3 questions. So I'll open up with a question if anybody else wants to ask anything. I'm sure that they'd be open to answering just about anything from the basic stuff to the very complex stuff. So my question is, in cardiomyopathy, you frequently develop, end-stage disease that becomes irreversible, which is quite a bit different than people with ion channelopathies, where you don't get a bunch of fibrosis and scar. And I just always wonder a bit about these cardiomyopathy gene therapies and the timing of intervention and reversal of the disease. And I see it much more as a condition that maybe is better reversed in the early stages. So if you can maybe both provide a 30-second response to that in terms of your plans and thoughts on inclusion in studies.
Sure. I think you summarized it very well. There's a Thai proverb. It's much easier to help an elephant that is getting up off the ground than stop one that's about to fall into the ground. We think that really across a large range of disorders, the goal is to try to get the right gene into the right group of cells before there's irreparable damage. And I think our Danon experience emphasizes that once you have decompensation, extensive fibrosis, even the most well-constructed targeted therapy is not going to be able to rescue that situation. Early diagnosis makes a great deal of difference in many of these diseases. And I think, fortunately, we are one of many academic and biopharma groups that are really advocating to enable more accurate early diagnosis. So many patients end up seeing multiple clinicians and caregivers before a correct diagnosis is made. That's true regardless of the genetic disease. And that's something that we can change. I think that's something that is changing.
I think that captures it all. I mean, that is the tricky spot here at gene therapies with patients is when to initiate. I agree with what Dr. Schwartz said. I don't know if I had much to add. I think it's the fibrosis and the scar. You're not reversing. In our animal models, we dosed with once they had overt cardiomyopathy, or we treated before they had an onset of disease. In both those cases, we would see some reversal, but it comes to a certain point.
I have a very basic question, but maybe a little bit unfair. I'm just wondering how confident are you in long-term expression of those genes once they're in there?
I'll take a stab at it. I mean, we know from other therapeutic areas, as I mentioned, we have efficacy out to, several years, over 15 years. I think the advantage in targeting cardiomyocytes, non-dividing cells after about 1 month of age really gives us a strong position for durability of effect. Dr. Schwartz demonstrated some efficacy out to 3 years in that initial cohort. So I personally am feeling pretty confident about the durability, and I'll let Dr. Schwartz add to it.
Yeah, well, certainly the cardiomyocytes are the heart is a good organ to target with AAV because even though there's non-integration of the genes, that epigenetic or episomal transduction is likely to last. Most cardiomyocytes, by and large, are not dividing after about 1 month of age. So whether you're treating adults or children, I think there's a very good prospect of long-term benefit. And we continue to see increasing evidence in the hemophilia world for the longevity of benefit with most, if not all, of the programs. In fact, just last week in Blood, one of the major hematology journal, there was a really nice update from a multi-country group with now 12 years of follow-up in a hemophilia dog model, showing that the durability of hemophilia therapy, and that's in the liver. And we know that liver cells do divide much more so than heart cells, so that gives us a lot of cause for optimism.
Great, thanks. And I think Dr. James, our keynote speaker, has a question. Maybe a comment is better in terms of timing, but yes.
I have actually a very simple and fast question. We've talked to a lot of patients about gene therapy trials on the horizon. The phase 1 trials are appropriately small and appropriately have relatively low-risk patients, but are complex and obviously risky. Our patients have a lot of uncertainty about whether or not to participate. Can I just ask very clearly, how is enrollment going in your HCM and ARVC phase 1 trials? It's different, right, than for Danon disease where the course is going to be bad.
Great, thanks. And this will be our last question, so hopefully we can keep our responses quick.
We haven't started our first patient dose in PKP2, so that's that question answer. And we've screened. We did a phase 0 seroprevalence study in our HCM cohort where we enrolled over 100 patients just to get antibody titers outside of the screening window. So there's a lot of enthusiasm. These trials are rigorous, and so I think with the hospitalization requirement, if these patients have jobs and children, it's very demanding in the first probably 3 to 6 months. So it does make it more challenging. We are going to release some data at the end of this year, so I can't spoil that news. But it is more challenging, I think, because of the rigor of the clinical trials.
That's very well stated. And I can state that for the PKP2 ARVC study, enrollment is happening. We'll provide more granular updates potentially later this year or soon thereafter. because of SEC requirements and all sorts of other stuff that I'd rather not even mention here. I can't really provide the things. You don't want me to go to jail for insider trading, because that will make enrollment that much more difficult. More on that during lunch.
I'm just happy to hear the answer isn't 0.
Great, thanks everyone. We'll move on to the next session.
speakers? You want us? Oh, okay. Ah, here we are. Great.
So we're going to try to start this session at 1:10. Each speaker gets about 8 minutes.
Yeah.
And then about 10 minutes per session. Sound good?
Sounds good.
We've done such a good job of staying on
Robert, like, it was a nice talk, but he was supposed to, I think it was like 10 minutes. It was like 35 minutes long.
I'm going to skip through a lot of my slides in my talk.
What do you mean? Oh, no, yours? Oh. Wait, you have 10 minutes?
I'm going to be 10 minutes.
Okay, good.
I have a lot of background that I can not use. It's just present.
Hey, how are you?
Good to see you again.
Good to see you.
I know Megan touched base about that 15-year-old that showed up in our merge. There's 260, they shocked her 4 or 5 times. She got up. Anyway, I looked at all the stuff. I said, it smells like ARC to me. I think they described it all to you and they said exactly the same thing. So I said, I can't send this girl home without a device.
Did it?
We were sure because she was a lead gymnast since the age of 3. So intensity and probably about
Is her arrhythmia suppressed enough that she won't get shocked?
well, so in the hospital just on beta blockers and nothing. The device is 6 weeks after the device we need to come back with one appropriate shot. So we redesigned it some ACP and switched her over to beta blocker and so on. See how she does. Otherwise normal. Well, apart from her biggest function but not a lot of
One of our former fellows is in Costa Rica and has a boy with age 5. It's like crazy young. Yeah. And then Dr. Hoga has some young patients as well. She wrote that paper about the under 12 presentations. Yeah, fairly. Got 2 more times. Yeah, October and November. You're going to hire somebody. I think they do. I think they do have somebody. Yeah.
Can we, oh great. So could everybody please get seated for this afternoon? So as mentioned, we're going to be more strict with the time this afternoon so that we can hopefully finish on time as well and have time for discussion. So if everybody could please stop talking and we'll have, if the speakers for this session could come up and we have the moderators here now and we will start the afternoon session. So thanks very much and I'll hand it over to Dr. Hamilton.
Good morning. You guys behave so well for Dr. Roberts. It's my pleasure to be here at this excellent meeting, and I have the pleasure of introducing Dr. Samya Das, who's both an MD and a PhD. He's Associate Professor of Medicine at Harvard and Director of the Resync and Advanced Therapy Program at Mass Gen. Runs a nice basic science lab himself there, looking at things like biomarkers, which really interests me. But he's made the translational step to be co-founder and principal scientific and medical advisor for Thrive Therapeutics. Welcome.
Thank you very much for that kind introduction, and thanks to the organizers for having us. Thrive Therapeutics, which spun out of work I'd done about 15 years ago, is also proud to kind of help sponsor this meeting, so really happy to be here and share some of the work. These are my disclosures, and of course there is a conflict because I founded Thrive and I consult for them, but so what you'll hear, take that with a grain of salt, I guess. So I wanted to talk a little bit about the science behind what we are trying to do. This is, I think, a unique approach to treat arrhythmias. And it started with work looking at a kinase called SGK1, serum and glucocorticoid responsive kinase. And this is a somewhat unique kinase because at least in the heart, it is activated in conditions of stress. And I know people don't like to see Western blots and things, but you can see the one below, that's from human patients. And you can see that at baseline, the levels are quite low of the phosphorylated form, that's the active form of the kinase. But in stress conditions like heart failure or hypertrophy, it gets markedly upregulated. And so this sort of led to a series of experiments that was published in 2012 that led to design of mice where you could activate this kinase and study what it does, or inhibit the kinase and see whether there was some effect, some beneficial effect of inhibition. And you can see that we designed these mice quite well. You could get an increase in activity of these kinase in the hearts of these mice specifically. And when you looked at these mice that had activation of the kinase on the right-hand side, you can see ECGs from these mice. On the top is a wild-type control mice, and on the bottom is a mouse with activation of this kinase. These mice were really prone to arrhythmias. And mice generally don't have ventricular arrhythmias, so that was somewhat unusual. These mice, when you stress them, either by putting catheters in the heart and stimulating them or subjecting them to heart attacks, they developed these intractable arrhythmias and even died. And you can see that on the quantification on the graph next to it. And interestingly, when you inhibited these kinases, the opposite happened, so that when you stress them, they actually didn't have these arrhythmias and did quite well. And if you took heart cells from these, so those are action potential tracings, these are heart cells that were taken from these mice, even to the layperson, you'll appreciate that on the right side, which is the cardiomyocyte heart cell with activation of this kinase, the action potential is quite a bit broader. It's longer. So there's a prolongation of the action potential duration. And in fact, if you look at these cardiomyocytes long enough with patch clamping, you can see they get these spontaneous activity that's shown below. And these are what really lead to arrhythmias in patients who have these prolonged action potential durations. And so we next sought to understand why that was. And interestingly, it appears that there's a channel called the sodium channel that's important in depolarizing the cardiomyocytes. But if these channels don't close, you get a persistent sodium current, and that's called the late sodium current, quite important in conditions like long QT syndrome, particularly long QT3. And these cardiomyocytes all had about 4-fold increase in the late sodium current. And we went on to show that this was due to the fact that these channels get phosphorylated by this kinase that, again, is activated in conditions of stress. And we could block that late sodium current. You can see that in the two graphs below. And if you block the late sodium current, you could not only normalize the action potential duration, but you could get rid of these extra beats that you see that lead to these arrhythmias. So we had pretty good data that inhibiting this kinase blocked the late sodium current and decreased this arrhythmic potential when this kinase is activated. And so this sort of led to, spam call. So this sort of led to a hypothesis that perhaps inhibiting this kinase might be important for those conditions where there's an increase in late sodium current. And this really led us to this long QT syndrome. We all know about long QT syndrome. We see patients with them. There are 3 major types. I'll really talk about long QT 2 and 3 because those are the 2 types where there's actually an increase in the late sodium current. And this is just a graphic of what that looks like. And you can see that the QT interval, which is the surface ECG marker that corresponds to the action potential duration, is prolonged in these patients. And the corollary from this is that perhaps if we shorten that QT interval, there's pretty good epidemiological data that the longer your QT interval, the higher the risk of arrhythmias. If you shorten these QT intervals, that you would decrease this arrhythmic risk. So that's what the whole premise was based on. And in fact, if you take patients with long QT3 and you derive from their stem cells, you can derive iPSC, induced pluripotent stem cell-derived cardiomyocytes. They have prolonged action potential duration. And when you inhibit the kinase with a tool compound that we developed, you can shorten the action potential duration. And we went on to show that you can actually decrease these extra depolarizations, these triggers of arrhythmias. And so this was really the basis for formation of what used to be called Long QT Therapeutics and then became Thrive Therapeutics. It was formed in 2019 with the usage IP from Beth Israel Deaconess, which is where I was at that time. And we were able to acquire a really potent series of SGK1 inhibitors from Sanofi and develop those into much more specific inhibitors with much less off-target effects. The company's done quite well with the Series A completed. And as I'll show you, we have some interesting results, both preclinically and clinically. So one of the first things we did, we talked to Mike Ackerman, who has a huge collection of these cardiac cells derived from patients with long QT1 and 2 and 3. And he tested these compounds that we developed to see that if we could actually shorten the action potential duration. And initially, we were really focused on long QT3, but from Mike's work and from work done with others in Europe, we found that not only in long QT3, but also in long QT2, you could actually shorten the action potential duration quite consistently with these inhibitors of SGK1. And in some cases, you could even do it in long QT1, although we've not quite focused on that. This is work done with a consortium that we work with in Europe, and these are various different models that are shown here. This on top is an iPSC cardiomyocyte model from patients with long QT2. You can see that again, we can shorten the action potential duration. We can get rid of these after depolarizations. This has now been done in 3 or 4 different families of long QT2, and it seems agnostic to the type of mutation that's there in long QT2, so that's good news. On the right-hand side is a rabbit model of long QT2. You can appreciate the really long action potential that's seen in these rabbits. When you treat these rabbits with the inhibitor, you can, again, pretty much normalize the action potential duration. And the last 2 graphs show a different approach. So obviously, this is an orphan disease, hard to recruit patients. And we thought, well, if you're seeing it in all kinds of long QT2, what if we pharmacologically block the channel that's responsible for long QT2, which is the HiRO channel? So you can do that with a drug that's approved. We use it all the time to treat patients with atrial fibrillation called dofetilide, very specific blocker. You can do that, and you can prolong the action potential duration. And when you treat the cardiomyocytes or even the heart, so below is a guinea pig ex vivo perfused heart treated with dofetilide, prolongs the action potential, you treat it with an SGK inhibitor, and you can normalize the action potential. So a sort of pharmacological model of congenital long QT. And you can do this even in an in vivo model. So there's a canine model, anesthetized, treated with dofetilide. You increase the action potential duration. And when you treat these dogs in a short period of time with long QT inhibitor, you can, again, normalize the action potential duration. So this led to our first sort of proof of concept trial in patients. These were healthy volunteers who were brought in, treated with dofetilide, and then we treated them with the SGK1 inhibitor to see all the things we had seen in guinea pigs and dogs, that when we shortened their action potential duration, did we see that here as well? And in fact, so this is the study design. In the interest of time, I won't go over it in great detail, but each patient served as their own control. It's a thorough QT type study where patients were treated with dofetilide and the drug, washed out, and then just treated with dofetilide, so we could compare apples to apples, right? And this was done in 2 different ways. So either they had their placebo first and then the then the SGK1 inhibitor, or vice versa. So this is what those results showed. So if you took all patients, and one of the caveats of this study was that because we couldn't really give patients the drug to prolong it past 500 milliseconds, that's what the FDA approval is based on. We didn't have patients with really prolonged QT intervals. But what we saw is if you take all comers, we could shorten the action potential duration by about 10 or 15 milliseconds, depending on how you slice the data. And in fact, if you took the patients with the longer QT intervals, you could shorten it even more, which was quite along the lines of what we were thinking, that you want a drug that is much more effective and your QT is much longer. If you take, and we'd previously done a study in normal people without dofetilide where we didn't overshorten, that was the other concern. So this was all good news. And if you, in fact, slice it by looking at each of those patients, you can see that those patients who had really prolonged QT intervals would shorten their QT by 40, 50 milliseconds, which was really quite meaningful. And those that had somewhat lower did so to a less degree. But the important point to note is that all those patients who were placed on the SGK1 inhibitor had QT intervals after treatment that were in a range that we wouldn't consider to be at a high arrhythmic risk. So having done that, we are now soon to actually look at results in patients, about 12 patients, proof of concept study with congenital long QT syndrome, long QT2 and 3, that have been treated with 2 different doses of this inhibitors, a short duration study, about 4 days, with an endpoint looking at their QTC. And in all these patients, their QTC is more than 480, which is where we consider them to be at a high arrhythmic risk. And so, what we're going to hopefully unveil soon is whether the drug works in shortening their QT interval. So I'm, in the interest of time, I think I'm good, right? Good. So I'm going to stop there. We have a couple of other programs for atrial fibrillation and HFpEF, where interestingly, SGK1 also is activated, but I'm not going to talk about that, given this is an inherited arrhythmia symposium. And thank you very much.
Excellent. Well, next up, we have Dr. Howard Sirks. He's the Chief Medical and Scientific Officer at Cardurian Pharmaceuticals. He's a physician scientist and cardiologist who ran a basic cardiovascular lab at Tufts Medical Center in Boston before spending the last 15 years in pharmaceutical drug development. So welcome, Dr. Sirks.
Thank you very much. I really would like to thank the organizers of the meeting for inviting us here. It's both exciting and humbling to be here amongst people who are really on the forefront of treating patients with inherited arrhythmic disorders, and of course to be here with patients as well. So we really appreciate it. So I'd like to spend a few minutes telling you about our company and then focusing on one of our clinical programs, which is a small molecule inhibitor of calcium calmodulin-dependent protein kinase 2 for the treatment of catecholaminergic polymorphic VT. So Cardurian is a company based in Boston that has 2 clinical stage programs. We have an inhibitor of phosphodiesterase 9, which is currently in clinical trials for heart failure. And as I mentioned, we have a CAMK2 inhibitor, which we are exploring for multiple CV indications, including CPVT, and that's what I'm going to spend most of the time talking about. We are really focused on exclusively new drugs for cardiovascular diseases. The PDE9 inhibitor is currently in phase 2 proof of concept trials, and a CAMK2 inhibitor, as I'll describe later, is just initiated an initial trial in patients with CPVT. This is just a snapshot of our pipeline. As you can see, our most advanced program right now is our PDE9 inhibitor, and we have really focused a lot of our research activities on developing small molecule CAMK2 inhibitors. As I'll describe in a moment, this is a target, CAMK2, that's been pursued in the pharmaceutical industry for a long time, and we've, through just deploying a lot of force and medicinal chemists and biologists at this target for a number of years, have had some success on getting molecules that can be brought into clinical trials. And so we have both oral and intravenous inhibitors that we are developing for a range of cardiovascular disorders. So focusing in on the CAMK2 inhibitor program, my apologies to the electrophysiologists in the room for going through excitation-contraction coupling, but for everyone listening, and just to also put into context why CAMK2 is so interesting, I'll just spend a moment on this. We know, as everyone knows, depolarization of the cardiac cell membrane leads to opening of voltage-gated calcium channels and an influx of calcium into the myoplasm, and that calcium binds to and phosphorylates, that calcium binds to and opens the ryanodine receptor at step 2 on the slide, leading to a large amount of calcium flooding from the sarcoplasmic reticulum into the cytoplasm. That calcium binds to the troponin complex on the actin-myosin filaments, leading to actin-myosin cross-bridge cycling and contraction. In cardiac relaxation, that calcium has to be removed from the cytoplasm, either by actively pumping it back into the SR or pumping it out of the cell in exchange for sodium. Now, CAMK2 is a protein kinase. Its function is to phosphorylate its target proteins, changing their properties. And CAMK2, as the name would suggest, is a calcium-sensitive kinase. When the calcium levels rise during the contraction phase of the cardiac cycle, CAMK2 is activated. And when the calcium levels fall, CAMK2 becomes relatively inactivated. And CAMK2 can become hyperactivated in the setting of multiple diseases. And that generally occurs through post-translational modifications to the enzyme. And that can lead to disease because CAMK2 phosphorylates multiple proteins, many of them ion channels, in the cardiomyocyte. And that can lead, and when CAMK2 is excessively active or no longer calcium responsive, that can lead to inappropriate calcium conductance and arrhythmias. Importantly for CPVT, the catecholamine surge that occurs with exercise or emotional stress may activate CAMK2, and that may be a factor that could precipitate arrhythmias in patients with CPVT. That has been demonstrated in some animal models, which I'll mention in a moment, and is what we are currently investigating in CPVT patients. So CAMK2 has been a difficult target. As I mentioned, it's been sought after. CAMK2 inhibitors have been sought after for a number of years because there are so many non-clinical models that have demonstrated an important role in disease states. And we are fortunate to have the first one that has gone into human testing. And as I mentioned earlier, we are developing both oral and intravenous forms for different diseases. One of the reasons it's been so difficult to develop CAMK2 inhibitors is with most kinase inhibitors, the trick is to find a molecule that is sufficiently specific for that target and doesn't inhibit other targets. And of course, potency, so that a reasonable administered dose can have the appropriate effect on the molecule. And very importantly for CAMK2, it's important that the molecule not get into the central nervous system because CAMK2 plays a very important role in the brain in learning and memory. So one of the central tenets of our discovery process, our research process, has been to only focus on molecules where we can demonstrate that they are excluded from the brain. And this is just one example where in the figure on the left you can see the plasma exposures in the clear bar and the minimal exposures in brain tissues to the right. And that's at a dose that is many-fold higher than what we administer to humans. So when in developing CAMK2 inhibitors for CPVT, we are really standing on the shoulders of giants. The role of CAMK2 in CPVT has been demonstrated by others, going back to Silvia Priori's study back in 2011, shown on the left, where she showed in a ryanodine receptor mutation knock-in mouse model that KN93, a CAMK2 inhibitor, could prevent the bidirectional ventricular tachycardia induced by epinephrine and caffeine. And more recently by Vasilios Bezaridis and Bill Pugh. Dominic Abrams, by the way, was on that paper as well, in Circulation, which showed using a AAV transduced peptide inhibitor of CAMK2 that they could also prevent catecholamine-induced or programmed electrical stimulation-induced ventricular arrhythmias with CAMK2 inhibition. So we have now initiated studies with our specific and potent CAMK2 inhibitors. This figure just shows an example of the pharmacokinetics and how we can establish CAMK2 inhibitory activity in humans. On the left are the plasma concentrations, a lower and a higher concentration, and on the right are the associated levels of CAMK2 inhibition that we have detected using a novel CAMK2 activity assay from the blood, where you can see we can get down to almost complete inhibition of CAMK2 activity. And so we have now embarked on a clinical trial in patients with CPVT. We are doing that in collaboration with investigators who are in this room, who we are very grateful to for their advice, and also CPVT patients who we are also very grateful to, who have given us an idea of what it's like living with this disease, and also what would be feasible to do in terms of a clinical trial. And so the concept is an initial single-dose study. It's an intrapatient dose escalation. And these are in patients who have had a treadmill test that demonstrates that despite their standard of care therapy, whether it's beta blockers, flecainide, sympathetic denervation, that they still have some ventricular arrhythmic activity detectable. And then we are randomizing them to one of two sequences where each patient has 3 more exercise treadmill tests. Before each test, they either receive a placebo or one of 2 dose levels of the CAMK2 inhibitor. The overall schematic is shown here. Our initial trial is in 12 patients, initially in adult patients with CPVT. We are starting our investigation in patients who have known RYR2 mutations. It is quite possible that CAMK2 inhibition may demonstrate efficacy in other CPVT mutations, but at least in the initial study, we are beginning with a slightly more specific population, although RYR mutations are already a very diverse group. And we are conducting the trial at 11 sites in the US, Canada, Italy, and France. And then the schematic is, as you can see here, those are the patients who in screening who have demonstrable ventricular arrhythmic activity are randomized to one of 2 schedules where they come in 3 times for treadmill tests about a week apart, and each time they receive either dose A or dose B or dose C, which could be 2 dose levels of the inhibitor or placebo. And the main endpoint in the trial is the level of inhibition of exercise-induced ventricular arrhythmic activity. So just to summarize, we're excited to have the first CAMK2 inhibitor in human clinical development, and very eager to determine whether this molecule can potentially bring some help to patients with CPVT. We've been assessing its safety profile in phase 1 healthy volunteers. And we now have also demonstrated that we can inhibit CAMK2 robustly in humans. And our first trial in patients with CPVT is now underway. We have an optimized formulation of this molecule that we think that is clearly giving us good concentrations in the blood, and is something that is easy to take, and we can bring all the way through development and into patient use if it is successful. And of course, we are considering other indications where CAMK2 inhibition may be able to confer some benefit to patients. So again, I really want to thank everyone for inviting us here. We're really honored to be able to present to this group and participate in the symposium today. Thank you.
Thank you both for the excellent talks. In the spirit of today, we are over time, but we probably have time for we're okay? Okay. Perfect. Any questions for our speakers?
Don't be shy. There are all these questions this morning. You're over time. There are questions now.
I guess I'll ask a question. So in terms of the CPVT program, you mentioned that patients will need a pathogenic RYR2 variant. Now, we're always plagued by the dreaded VUS. So in patients who have a clear diagnosis, will they be eligible for your study?
Thanks. So initially, patients in this first 12-patient trial will require a clear known mutation that is associated with clinical CPVT. And that is really just because it's our very first study, and we want to begin with a very clear-cut population. But I do think that there is potential for patients with the clinical syndrome and VUS mutations, as you noted, to benefit. And we hope that if we can demonstrate some activity in the initial population, that we will broaden it to include those patients you mentioned.
I just had a quick question. I was curious about the SGK1 inhibition, and it seemed to work in a bunch of different arenas, long QT1 and 2 and 3, and it all had the same effect. So why is it that is it just an effect on that sodium inward current? And is the secondary effect through sodium-calcium exchange where you decrease that current as well because there's less sodium inside the cell? Is that why it works in all three forms of
Yeah,
QT?
so it works. The most robust data we have is in long QT2 and 3. Long QT3 is pretty obvious because there is a late sodium current, and I think it changes the channel biophysics of these channels and prevents that. Long QT2 is actually interesting. We just, the hypothesis has been when you prolong the action potential duration, as you point out, you have more sodium influx, and that secondarily leads to more calcium. In fact, there's actually pretty good data that even in long QT2, there's a large amount of late sodium current. For example, for these patients who have really prolonged QT interval in long QT2, you can treat them with mixilotine. More and more people are doing that, and you can shorten their action potential duration. You can decrease their arrhythmic risk. And now we have direct data that if you take, for example, the rabbit model or these iPSC cells with long QT2, they have a substantial increase in the late sodium current, and inhibiting SGK1 normalizes that. And so it doesn't act on these potassium channels, but what we think is happening is that actually CAMP kinase 2 gets activated in these. In fact, there's good data that when you prolong the action potential duration either in long QT2 or even treating with dofetilide, you can actually activate CAMP kinase 2. And SGK1 actually happens to be a substrate of CAMP kinase 2. And so we've shown activation of SGK1. We think secondarily it might be through CAMP kinase 2. We haven't quite proved that yet. There are some experiments undergoing to prove it. But I think that's why SGK1 gets activated, and that's why it works. Why it doesn't work in long QT1 is not totally clear to me. And it works in some, there appears to be a mutation-specific phenotype there. But if someone has any ideas why it doesn't, I'd be curious to know.
Would sodium-calcium exchange inhibitors do the same thing as SGK1?
So the sodium-calcium exchangers would probably work upstream of that, right? So you could imagine that if you block this reverse mode, then you may not get activation of CAMP kinase 2, and maybe you get less late sodium current in these conditions other than long QT3. I think people in the past, I think someone from UCLA had really developed this to some degree. I'm forgetting his name now. But for some reason, they never took off in trials just because I think you'd have to inhibit that reverse mode of sodium-calcium exchange. And I'm not an expert on this, so somebody else might know. But those inhibitors were never, they worked fine in cell culture, but in vivo, there was always some issues. The genetic models were fairly convincing, I think, when they tried that. But I think for whatever reason, that there weren't any good inhibitors to inhibit that specific mode of sodium-calcium exchange.
Thanks.
I have a quick question for both of you. So thanks so much, and I think it's great that there's targeted therapies being developed for CPVT and long QT. I remember when I was a medical student, I learned about these really elegant physiology, and I was like, we treat them with beta blockers? But with that said, beta blockers for long QT and beta blockers plus flecainide in CPVT work pretty well. In terms of what clinical trials do you guys envision doing in the future to show the world that we really need to be treating our patients with these new targeted therapies instead of the other medications that literally cost dollars a month? Have you guys thought forward about that?
Yeah, that's a great question. So as you said, beta blockers and flecainide are pretty cost-effective for CPVT, and a lot of patients have good suppression of ventricular arrhythmic activity with those drugs. So we are targeting, at least for now, patients who don't have extremely good control on those medications. So it is a subgroup who we think still have unmet medical need. They're having events where they're having ectopy demonstrated on their treadmill tests at an unacceptable level, and there's a desire for more control of that. And our initial approach is to see whether CAM-K2 inhibition will provide more prevention on top of that, on top of those therapies, not in place of. But I think if that is demonstrated, the next approach would be to see whether a CAM-K2 inhibitor on its own would be better than those established therapies. And that would have to be very carefully done after demonstrating efficacy on top of those therapies in some sort of a withdrawal trial that we could envision in the future. But the initial approach is to not replace these drugs that are used and inexpensive, but to target the patients who, despite them, are still at risk.
Yeah, I mean, I think for long QT, there are a couple of arguments. One is the beta blockers work really well for long QT1, maybe modestly for long QT2, and not so well for long QT3. So long QT2 and 3 are the target we're first using. The second is, I mean, probably a lot of you in the audience have patients, young patients on beta blockers. And I can tell you that I have patients who even had syncope who refused to take beta blockers. So there's a compliance issue. I think there was a nice study from New Zealand, I think, that looked at this compliance and in patients with long QT, and it's somewhere in the 40% range, 40% non-compliance. So, and there is actually good data that when people come off the beta blocker, their arrhythmic risk is even higher. So I think if you could have a drug that doesn't have those side effects of beta blockers, and for all we know right now in the patients that we have dosed, they've really had no side effects on these drugs. And in fact, some of them have volunteered to come back for dose escalation studies. So I think that would be the argument. For long QT1, I think it's a trickier argument because beta blockers do work pretty well. But again, I think you could target some of those patients. For example, there are patients who are young kids, especially with sympathectomy, who are, so would this be a better approach than sympathectomy? I would argue that maybe so, if it's safe and it's durable. But that's sort of, for now, we all the patients who have been dosed have been on a beta blocker, apart from 2 who couldn't tolerate it. So the initial trial will be on patients with beta blockers.
Thank you, everybody. I think with that, we'll close. Pam, do you have a burning question?
Well, patient perspective, yeah. Perhaps. What kind of commitment does a patient need? If I joined the what kind of commitment do I have to make as a patient? Is it over a long period of time? Is it one-time deal?
Yeah, it's a great question. It is over a number of weeks. It's not an extremely long period of time, but it's not by any means an insignificant commitment, because it does require several exercise treadmill tests and to come back to the clinic each time to have them performed. So it is, three treadmill test visits after an initial screening visit. And so, we recognize that's a lot for patients, and we're always looking for ways to make it easier, make it more comfortable, and it's a big focus on what we try to do, but it is a substantial commitment, we acknowledge.
Thank you, everybody.
could we turn the? Okay, great. Great. Okay, so we've got to keep things moving, and I'm not trying to be the bad guy, but if we don't keep things moving, we're going to finish after 6. So, yeah. Great. Okay, everybody. So this is our second gene therapy session. So I'll hand it over to the moderators. Thank you very much, everybody.
Hello. So I'm here to introduce Dr. Gabriel Brooks. He is a trained clinical cardiologist and the Chief Medical Officer of Solid Biosciences, a leading precision medicines company for neuromuscular and cardiovascular diseases. Gabriel has been involved in the development of medicines for rare cardiovascular diseases, from pulmonary hypertension, disease gene cardiomyopathies, Anderson Fabry, and long QT, including leading studies in late sodium channel inhibition through eleclazine. He's been dedicated to gene therapy development for the last 10 years, leading development at 4D Molecular Therapeutics, and then leading rare cardiovascular disease development at Pfizer before moving on to Solid Biosciences. So welcome, Dr. Brooks.
Thank you, and thank you, Jason, for letting people have a break. Otherwise, instead of the break, you would have had me, which is probably not a good thing for me. Really a privilege to be here, and just really stunning to have this day where there is so much interest in precision medicines for rare inherited arrhythmia syndromes and arrhythmia in general. So we can probably go to the next. Oh, I need the clicker. There we go. Otherwise, I'm going to say next slide. So there may be some forward-looking statements, but one statement I'll make is that we're in a quiet period with some of our package under review, so I'm not going to be able to share too much data about our CPVT gene therapy program or clinical plans. So, Jason, that may mean that we can make up some time. To start with, I'd like to start with my acknowledgement slide first. Really, I want to acknowledge the extremely strong role that patient advocacy should really play, and certainly does play at Solid in terms of making sure that we know what our North Star is. So thank you to Canadian SADS, to the SAD Foundation, and thank you for HiRO, I think, for having the foresight of bringing translational medicine into this venue. I'd like to thank our lead clinical scientist, our lead scientist, non-clinical scientist, Elena Bresnow, who's done a lot of the work here, and our collaborators at Mount Gary. And then thank you to all of you in the room. So Solid Biosciences, you may not be familiar with the company, so let me just very briefly say this is a company that has been in the gene therapy space now for many years and started in Duchenne muscular dystrophy, where it was a single disease, single therapeutic company. And through that, we actually have very quite experienced leadership within the gene therapy space. We've been very collaborative in terms of reaching out to probably many of those people in this room. We have a diversified pipeline not only for Duchenne muscular dystrophy, but now a robust cardiac pipeline. And then really having focused on high-dose gene therapy in the Duchenne muscular dystrophy space, we've had to really focus on how to do manufacturing for AAV. And that's critically important, and especially in places that are cost constrained. Our goal is really to be able to drive down the cost of goods for AAV, which hopefully can democratize these precision medicines. And that's really, again, I think really important for healthcare systems that are cost constrained. So in terms of our pipeline, as I alluded, we have been in the gene therapy space for Duchenne muscular dystrophy with now a discontinued program. We're back in the clinic with a next generation gene therapy for Duchenne muscular dystrophy. And this is really an interesting space. This is a place where the regulatory framework is very fluid, and we're seeing that approvals of the gene therapies with a different pathway of traditional approval. And so I think it's really good for the community that we're all learning from how we can think about accelerated approval and confirmatory packages. In addition, as I alluded, we have a really robust cardiac pipeline, and our lead program is for CPVT. So really what's fundamental for Solid Biosciences is the proposition that precision medicines that closely target the pathophysiology of disease are likely to be more successful in terms of actual clinical benefit. And so, we are targeting CPVT, and from our perspective, this is a disease that affects young. It can be highly malignant, and historically has had a mortality rate that has been quite high. And we can see on the lower left part of the slide, there are effective therapies in the clinic, flecainide and beta blockers, as we know, but we do see that they have breakthroughs, and I think that this is really, in terms of the proposition of a gene therapy for CPVT, where we sit. Why do they have breakthroughs? One of the things that we do know is that there are issues with compliance, especially in the youth, especially in adolescence. And when we talk to providers, when we talk to SADS, we understand that usually it's really in the younger population where there are issues. And so having a durable, continuous therapy is really consonant with what's important for these patients, so that if they miss a dose, they'll be protected. And that's the value proposition that we hope a gene therapy for CPVT can add. And so, just as Howard felt, I'm sure at the time, talking to a room full of electrophysiologists about calcium-induced calcium release is probably not the best way to show my expertise. But I'll just direct you to the RYR2 channel and the CALSA question, which are critically important in calcium-induced calcium release. And we know with CPVT, with beta-adrenergic stimulus, with catecholamine release, that we can have upregulation of CAM kinase 2 delta, as well as PKA, which can lead to increased loading of the sarcoplasmic reticulum, and that in the setting of either absence calsequestrin, CPVT2, or in the presence of gain of function in RYR2, that can lead to leak through the RYR2 channel and a trigger of delayed after depolarizations. So our approach to CPVT is one that actually, and I'll show you this in a couple of minutes, is one that addresses both CPVT1 and 2 by leading to overexpression of calsequestrin, which can increase the buffering in the sarcoplasmic reticulum, which can lead to a lower probability of the RYR2 channel when defective in diastole. So our approach is to use, again, a calsequestrin transgene in the setting of a muscle-specific promoter to lead to calsequestrin in the heart, and using a cardiac tropic vector. And in terms of our drug, which is called SGT-501, what is it that we see with this drug? So on the left-hand side of the slide, I'm just proving to you that we can actually express calsequestrin when there's no calsequestrin. When we do express calsequestrin in the setting of a calsequestrin null mouse, so a CPVT2 mouse, we can abolish the sustained VT even at very low levels of transduction, meaning we don't have to hit every single cardiomyocyte, even down to less than 33% of the cardiomyocytes. We can get an abolition of sustained VT, as well as affect NSVT and PVCs. So then, again, we actually can show that even down to very low doses, and here I'm not going to disclose what these actual doses are at this time, but I look forward to in the future. We can, in now an RYR2 knock-in mutant mouse, show complete abolition of the bidirectional VT across a dose range and 12-week-old animals. And then actually looking in the CPVT2 mouse, the knockout mouse, we can show that over time we can dramatically suppress the sustained VT, which is the manifestation of disease in that particular mouse model. And then in the lower right-hand box, you can see that actually when we look at transduction, we don't have to have complete transduction of the heart. We actually, if we have 40% of the heart or so cardiomyocytes transduced, we actually see this pretty profound pharmacodynamic effect, which is promising to us, and that's why we're really excited about the development. In terms of SGT-501, we have chosen to take a capsid that is cardiac tropic as well as, or as much so as some of the other cardiac tropic vectors. You might not be capsid biologist. AAV9 is kind of a workhorse capsid that's being used in many programs, and we know is cardiac tropic. RH74, it's RH because it was discovered in a monkey, also fairly cardiac tropic, and our vector is likewise cardiac tropic, except we're using a vector where we believe that the safety profile is superior. In terms of expression, we can see that we can lead to high expression of the particular transgene here, CASQ2, as high as other cardiac vectors. And then with this promoter, we have selectivity so that we demonstrate cardiac expression of CASQ2 in the heart, whereas we don't see it in the other muscles. So what is our clinical development strategy? Again, I'm not going to be walking you through the protocol at this time because we do have some dossier under review. But what we believe is that there's a general treatment paradigm for CPVT, which does vary by site and geography. But in general, what we aim to first start studying are patients that are high risk, those that are on beta blockers and flecainide who, similar to the approach discussed before, still have an evidence on exercise stress test of arrhythmia. But ultimately, we believe that if we can be successful in demonstrating protection, that we can then move into more of a preventative model where even we can start titrating patients off of their beta blockers and flecainide if they find those medicines onerous. And then how are we going to test the efficacy? So exercise stress test does, as a provocative measure of arrhythmia through the ventricular arrhythmia score, is certainly very attractive, as well as ambulatory monitoring for both safety and efficacy. And so then the next steps, so we are really pleased to share with you that this program is moving rapidly to an IND, which we anticipate opening early next year. We've already discussed with the the FDA, it's in discussion right now, the pre-IND, which I've alluded to a couple times now. And then we have the appropriate orphan drug designation both in the EU and the FDA. And we are having robust discussions with those regulatory agencies. And thank you.
Thank you, Dr. Brooks. Next up, we have Dr. Richie Khanna. Richie Khanna has 20 plus years of experience in leading comprehensive non-clinical development and clinical bioanalytical programs. Dr. Khanna joined Lexeo Therapeutics in 2021 as Vice President of Non-Clinical and Bioanalytical Development. She is mainly responsible for all the IND-enabling studies across all programs, as well as clinical bioanalytical assay development for all phase 1 studies. Prior to Lexeo, Dr. Khanna worked at Amicus Therapeutics for over 16 years. Dr. Khanna has a PhD in molecular biology and a postdoc in biomedical science. So welcome.
Thank you very much for the nice introduction. Good afternoon, everyone. It has been really exciting for me because this is my first opportunity to attend this HiRO Symposium. And I'm very excited to show what we have done in Lexeo. I'm someone, as you can make out from the introduction, I wear a non-clinical hat as well as clinical hat, which makes it even more passionate for me to work into what I do. So to start with, these are my disclosures. These are my disclosures and disclaimers. So before I go into PKP2, which is something the topic of interest for this audience, I just want to go through quickly the pipeline that we have in Lexeo Therapeutics, what we are actually very passionate about. Lexeo is mainly looking after the cardiovascular programs, but we do have CNS, especially Alzheimer's disease. So among the cardiac programs, the very first one is the FA cardiomyopathy. This is a program in which the gene of interest is frataxin. It's a mitochondrial protein. We are taking the gene therapy approach, and our platform capsid is AAVrh10. This program is right now in phase 1/2. We have completed the 2 cohorts dosing, and we are in the process of assembling all the data, and it should be releasing soon. The second program that we are working on is PKP2-ACM, which is very near and dear to this audience. This program, as you know, the gene of interest is PKP2. It is also in phase 1/2, and we are actively screening. The next program is also, so I forgot to mention the PKP2, as you know, is a desmosomal protein. The next program is also a desmosomal protein, DSP, that also we heard this morning. We are going with a slightly different technique for that. The gene of interest is connexin 43, and maybe some other time in the next symposium I can actually show you how we are approaching for that, an indirect approach using a connexin 43 gene of interest to DSP cardiomyopathy. This is at preclinical stages. We are planning and initiating IND-enabling studies. Last but not the least is the HCM using troponin I3. This is at preclinical stage. We are into candidate selection. And among Alzheimer's, just very quick, we are actually focused on ApoE4 homozygotes patients. This is ApoE lipoprotein, which predisposes the patients to early dementia and Alzheimer's. This is actually one program which was very much advanced at Lexeo. We have completed the dosing, and we are in the process of assembling the data. There are also some second and third generation, which is a modification for the first one, but those are all at preclinical stages. So with all this pipeline, especially like focused on the cardiovascular, the one that I'm going to focus in details is the PKP2-ACM. This audience knows about PKP2 disease mechanism, but just to give an idea, once again, to refresh our memories, the reason we picked PKP2, as you know, this is the most prevalent mutation. This is the most prevalent gene in which you see the mutations. As a result of the deficiency of the protein, the entire desmosomal complex, because it's a part of the desmosomal complex, is distorted. And we all know that in this disease, the patient's heart is fatty, it has scar tissues, the patients undergo arrhythmias, and unfortunately, the sudden cardiac death. We all know, and we had great discussions this morning about the ICDs, it is life-saving, but we all also know that it is not taking care of the underlying pathology, and moreover, the disease is progressing, right? So that's why there is a need, and I know there is a lot of players in this field, and we are also excited to be a part of it. So in Lexeo, the approach that we are taking, the gene therapy approach, is we have AAVrh10 as our capsid. We have tested it extensively. It's a remarkable capsid. We are using human healthy PKP2 transgene, and this construct, the transgene expression is mediated by a cardiac specific promoter. And we call this gene therapy as LX2020. You'll hear this name many times in my presentation. So the mechanism by which we think our gene therapy is working is with LX2020 administration, we are able to put the healthy gene, which restores the healthy protein, as a result of which, if you can see in the desmosomal complex, this is the bottom part that I'm talking about, there is a scaffolding of the entire desmosomal complex. And we believe because of that, there is improvement in cardiac morphology, structure, and cardiac function. So I'm actually going to show you the preclinical efficacy and safety studies, which I'm mainly responsible for before I enter into the clinical stage. So as you all know, that for any drug development, it is extremely important to have a very good mouse model. And Lexeo was very fortunate to actually collaborate with Dr. Farah Sheikh from UCSD, who has developed a novel PKP2 mouse model, which we were amazed to see that actually recapitulates all the hallmarks of PKP2-ACM disease in the patient. So this is a knock-in mouse model using CRISPR-Cas9 genome editing approach, and they had introduced the most common RNA splice site mutation into the PKP2 gene. As a result of this mutation, the homozygous mice that they obtained are extremely sick. As you can see, the red line is for the homozygous mice. They have a median half-life of about 11 weeks. But as soon as 4 weeks, you actually do see that these mice have sudden cardiac death. They die. The heterozygous mice is also having some phenotype, but not as dramatic as the homozygous. So what we have seen in these mice is with respect to cardiac function, if you look into the homozygous panel, the left and the right ventricles are dilated. As you know, that's a hallmark of the disease. And when we look into the function, the ejection fraction, this is the right panel, the red bars are the bars representing the mutant mice. The ejection fraction is significantly reduced. The ESV and EDV, which is the end systolic volume and end diastolic volume, are significantly increased. And if you look into the bottom panel, which is the electrical deficits, we see that the heart rate and PR interval is okay, but the QRS interval is widened. And with that widening, that is like the indicator of ventricular depolarization, which we all know is kind of a primer for arrhythmias. So the arrhythmias that you see in the patients are naturally seen in these mice. We don't have to introduce any kind of stress to get the arrhythmias. These mice, even as early as day 2, have arrhythmias, which was good for us so that we can follow the treatment. As you know, this is a desmosomal gene. With respect to, with the PKP2 levels being down, the entire desmosomal complex, DSP, DSG2, CHOP, not per se NCAD. When we tested, this is 4-week-old mice, but as the mice grows, some of those which are alive, they are extremely sick, even for NCAD, and connexin 43 levels are dramatically reduced. And finally, if you look into the morphology or pathology, the heart of these mice is extremely sick. When we do the H&E and trichrome staining, we see fibrosis, calcification, again, dilation of the ventricles, and extremely thin walls. So with a great model like this, it became easy for us to actually test our investigational drug, which is LX2020. We gave it to the mice. We had wild type, untreated, vehicle control, and we had 3 different dose levels, low, mid, and high. We chose to actually treat 3-week-old mice, which were already very advanced in their disease stage, which was good for us because we wanted to know whether we can rescue or potentially reverse. And we went up to 12 weeks to look into the treatment. So the first experiment was, of course, the PKP2 expression. And as you can see here, the green bar is the low dose, which was 6E12. It didn't do much. But the blue and the red were 2E13 and 6E13 respectively. And there was great increase in the expression over wild type, which was about 4-fold and 10-fold respectively in these mice. We were also able to see the blue and the red, which is, again, the mid and the high dose, the 2E13 and 6E13 respectively. It could reduce the preventricular contractions. We saw more mice with regular, normal rhythms as compared to the mice with the preventricular contractions. And though I didn't show over here, we did see reduction in the QRS interval. We did extensively MRI in these mice. That was one of the biggest things that we all know it's quite complicated, but we were able to do this, and we could see that there was a dose-dependent reduction in the EDV and ESV, both in the left and right ventricle, dose-dependent significant reduction. And with respect to ejection fraction also, we saw that it was going into the right direction. That is, the ejection fraction was increasing. So the bottom line for this was that with the increase in the expression, we saw the associated efficacy of improvement of the cardiac function. Moving along, as I mentioned, with the deficits that you see in the desmosome, we also found that with the PKP2 increase in the levels, we were able to increase the levels of money of the desmosomal and even gap junction markers that we saw were reduced in these mice. We also, of course, regardless to say, the survival was significantly improved. It was 100% both in high dose and mid dose. There were 2 mice that died very early on in high dose, and that was due to an injection error, but rest of the mice all survived, and we know the survival was very good at these doses. And finally, looking into the histological defects that we saw, if you look at the wild-type animals compared to the vehicles, they're extremely sick heart, but with LX2020, we could see a dose-dependent improvement. And some of the mice in the high dose, 6E13, actually looked indistinguishable from the wild type, which suggests that there may be a potential of reversal, at least in these preclinical studies. We then moved on to the safety, as obviously FDA wants to see both the efficacy and safety. And basically, we focused on to the 2 doses that I told you were very effective to make sure that they are safe in monkey studies. We went for 12 weeks. We did a lot of assays during this 12-week duration. There was no abnormality. Fortunately, this investigational drug in mice was extremely safe. And we were able to establish no EL. We looked into biodistribution and expression in heart. I'm just showing ventricles, but we looked into many different organs, and it was amazing. So I'm going to skip this because this is just a recapitulation of what I have shown, that we were able to improve all the hallmarks of the ACM that we saw in these mice. And that cleared our IND, and we started the first in-human studies, which is called as HeroiC PKP2. This is a 1-year-old, 52-week-old. It's an open-label study. It's going to be followed for 5 years. We, of course, are doing systemic IV administration. We are giving patients prophylactically the corticosteroid sirolimus as well as prednisolone. The key inclusion criteria is pretty similar, pretty easy. Anybody with 18 to 65 years of age can enroll the study as long as they have confirmed ACM, confirmed PKP2 genotype, they have ICD, as you know, it's important, and also minimal 500 or greater arrhythmia so that we can actually follow up. The 2 dose cohorts are the ones that we actually saw in our preclinical studies, very effective. That's what we are doing. Primary endpoint is safety, but we are doing a lot of other endpoints, arrhythmias, ECGs, echoes, MRI, cardiac biomarkers. We are also doing biopsies so we can look into expression and some of the things that I showed you in preclinical studies and the PRO. So my final slides, if you have any questions, we do have this site. We do have the information on the clinicaltrial.gov for patients, for caregivers, for medical affairs. If there are any questions, you can send us an email at lexioclinicaltrial.com site. And fortunately, I have my clinical lead colleague, Dr. Greg Aubert. I'm not a clinician. I do clinical assays and enable the studies, but if there are very complicated clinical questions, I have my colleagues who can answer. So thank you very much. Thank you for the organizers, patients, advisors for letting us do this study so we are at a stage and hoping the best for the treatment outcome. Thank you.
Jason, how much time do we have? -5?
5 to 10.
10 seconds of questions, 4-second answers. That was wonderful. Just while people are thinking of their questions, I just want to acknowledge the incredible pioneering spirit. I'm struck. I'm impressed that HiRO has come to such a translational place in a relatively short time. But many of our industry partners who have been in this field for quite a long time, I'm not going to try and age them, but they're up here and they say, well, I've been doing this for 15 years and 20 years and that. So just acknowledge that incredible vision that they had with these one molecule. I suppose it's like a marriage. You commit to one molecule and you just go with it, and then you just hope you've made a good choice. So the floor is open to questions. While people are coming up, let me ask you both just more of a philosophical question. Top 3 barriers to patient recruitment in each of your pathways ahead. You go ahead.
Again, maybe I think this is the best question for Greg. You want to address?
Sure. Yeah.
Do you want?
So while he's getting a mic, we'll
Yeah. So I think, having been in the gene therapy space for some time for cardiovascular disease, I think that the poor penetration of genetic testing, even when by guidelines, I will not get on my soapbox, I promise. But I think that's a huge barrier. So patient identification when we're targeting specific disease gene cardiomyopathies, or inherited arrhythmia syndromes, I think that's critically important. For AAV gene therapy, there's two huge barriers which we're all really working on. One is dosing patients that have natural immunity. That's really tough. That's kind of like going to the moon. The next one is redosing. We really hope that we're going to be able to make a meaningful, durable impact on patients with that one dose. But especially if in the Duchenne space, and we know that there's muscle turnover, not so much in the heart. If there's a prospect for if there's a prospect that you would need redosing right now, if natural immunity is like going to the moon, that's like going to Mars. And so there's a heck of a lot that we're learning about that, and that's incredibly important to understand. So those are two. I don't know if I have a third one. Think about it.
Yeah, no, that was a great answer. Yeah, I don't know if I have much more. I agree. Thanks.
Question from Dr. Cron.
So let's go big picture and talk about 10 or 20 years from now. So, genetic testing, I always make a joke and say genetic testing is cheaper than talking to the triage nurse because it's pretty accessible, right? So genetic information will be easy to get, and then the potential to fix something will be feasible. So how do you see development as we go through heel pricks at birth, school age clinical or genetic screening, access to curative strategies like we would do now for patients who have symptomatic WPW, for example, something like that. What does the future hold beyond the due diligence and process and safety efficacy language of right now as we're sort of emerging in phase 0-1? What do families have to look forward to? We've got some family members here who want to know whether their kids or grandkids will get fixed even though they may be too old to have it happen.
Maybe there are two parts to this question. The first one that you said, what do we need to do to maybe those newborn screening, if I understood your question correctly, that maybe in order to know whether you have the disease or not, the newborn screening that we used to do for other programs, we need to do this early on. And the second part was, I have to remind myself.
In a way, it's two points. One is, what's the right timing if we have a curative strategy and we have things like AAV intolerance and so on?
Yeah.
And then the second one is, does the future hold that this is almost like a genetic vaccination program?
So it's not going to be like if you have given to the parents, it's not going to go to the kids. It's not into the germline.
No, I don't mean that. I just mean your 80-year-olds are going to be seeking to use their payments or their estates to pay for their grandchildren's genetic vaccination.
If I could, I think so. I think partly baked into your question is the question of genotype phenotype, and we know, especially in some of the diseases discussed today, that there's going to be likely pathogenic patients that never manifest disease. Right. And so are we going to understand whatever the polygenetic risk or the environmental modifiers that are going to affect that person so that we could predictably say this person's going to have the disease, and therefore if we had a safe and effective therapy that was had low enough cost of goods that we could actually deliver it, who would you pick? I think that's huge. Getting there today, I don't think we're there. I think we're going to be compelled, I think, to start in patients that have manifest disease. And we've talked about not even just manifest disease, that they're going to have disease that has enough pathology that the value proposition, the risk benefit is worth it for an experimental genetic therapy today. So as I think, we hope that we can show that these therapies are safe, effective, we'll be able to dial that down and get into a less sick patient population. And then ultimately, if we were to do it as a preventative therapy, we would really have to understand exactly who's going to manifest disease. I mean, that's the logic that I have. I don't know if it addresses your question, but I think it addresses maybe the idea that you were elaborating.
I just wanted you to start talking about it because I think we have to think about how that's going to be. I mean, the cultural, ethical, economic aspects of implementing what we are talking about, if it's feasible, are massive. And it's not going to be something where we have a little Bethesda conference and call it good.
Absolutely. And it's not going to be with a, if we're getting into a preventative gene therapy, I mean, that's huge, but it's not going to be with a $3.5 million single dose, right? It's got to have a cost of goods that's way lower.
Maybe I'll just finish to build on Andrew's point, and speaking from the point of view of a government bureaucrat, which is what I am now, in addition to being a cardiologist. But as we go from the current state, which is something brand new, exciting, we all feel this is sort of a turning point, and we can envision a future that Andrew describes that's transformational. But we move now from very expensive, applied to rare diseases, very drug refractory, so a very small number of people, but there will be steps along the way until there's a sort of a more universal application. And one of the things that's always occurred to me is that people who do this kind of drug development, and it happens in the cancer drugs as well, is you get to the 11th hour and say, hey, we've suddenly got something, and payers and governments aren't ready to sort of enter this into their innovation pathways, which typically take years in their own right. So I'm just wondering at what point in your process you engage and say, listen, guys, we got something really big coming down the pipe. Let's start to talk about what the innovation pathway intake looks like. How can we accelerate the health technology assessment part of this? And then most importantly, accelerate the implementation, because nothing is worse for patients and families than having something that we know works and we can't get it into the field. Yeah. I don't know whether that invites a response or whether a retort.
I think that's food for thought.
Yes.
Okay.
Yes. Yes.
Thank you to our speakers, and we'll look forward to the next session.
That has been around for millions of years because bacteria use it in order to defend against viruses. And specifically, it can target exactly where you want to go, provided that you have created the right guide. So the guide specifically targets the certain portion that you want to edit. And we've developed really a modular system so that that circle part there, which is the 20 nucleotide 5' component, is modifiable. So we can target different genes depending on what those 20 first nucleotides composition is. These components fuse together within the cell, and you get this ribonucleotide protein, which actually does the editing. And we get an insertion or a deletion, which essentially knocks the gene out. People rightly ask, how do we develop really specificity and avoid off targets? This is gene editing after all. How do we ensure that we are being as potent as possible from the on-target perspective and avoiding off targets? And that's part of the really foundational work that the scientists and the research team does at Intellia. So we have to identify the right sites computationally in order to ensure a few different things. One is that we're avoiding sites that we don't want to get to, and two, that we're able to treat a broad spectrum of patients. So that means understanding what common SNPs there are and sequences that may prevent the binding of that ribonucleotide protein to the patient's DNA. So really important component. Then we do a lot of in vitro and cellular work in order to understand whether or not there are off targets that we might observe in patients, and to ensure that we have a potent therapeutic. So there are multiple orthogonal methods in order to establish this. And then we're going to put this into animal models in order to ensure that we get the appropriate knockdown that we're looking to achieve. And so this is, again, a tunable portion of the work that we do so that we can modify that 20 nucleotide 5' beginning in order to then ensure that different targets have the most potent knockdown with limited or no off targets. So I'll show that to you over here. This is from NTLA-2001, which is our ATTR cardiomyopathy program. And what we're looking at is the editing frequency of NTLA-2001 in primary human hepatocytes, as well as any potential off targets. And so what we see here, you can surmise, is that as we increase the dose or the concentration of our guide RNA, we do not see any meaningful off targets, indicating that we have a highly potent and selective therapy. And so with this data, along with standard preclinical models, we're able to take this into patients. How does the actual mechanism of action work? So NTLA-2001 is infused as a single dose and is taken up by hepatocytes. Because it's a lipid nanoparticle, it's taken up by hepatocytes and undergoes endocytosis. Within the LNP, you have the guide RNA, and actually the Cas9 component is mRNA, which is then translated into the Cas9 protein. Both of those are transient, as I mentioned before. In the hepatocyte, they form the ribonuclear protein, which translocates into the nucleus and edits the DNA at that target location, introducing indels and knocking out the gene. In this case, it's TTR for our lead program and KLKB1 for HAE. You probably know more about amyloidosis than I do, speaking to a room full of cardiologists. But essentially, this is caused by the accumulation of misfolded transthyretin protein. And actually has two manifestations, which you're probably familiar with, both the cardiac and the neuropathy components. Currently, there are agents available, but require chronic dosing. And as probably you've seen in some of your patients, they continue to progress, whether that's progression of their neuropathy or progression of their cardiomyopathy. And in many cases, this remains a fatal disorder. And patients are really looking for alternatives in order to potentially halt and even reverse disease. So our approach is to knock out the TTR gene with our single-dose CRISPR-Cas9 therapeutic protein. We can reduce the wild type and the mutant forms of TTR, and we can address both forms because this is a single gene disorder. From our perspective, we aim to halt and potentially reverse disease because we can achieve deep and consistent reductions in TTR. It's a single-dose therapy, and we expect this to be lifelong. And I don't show some of that data today, but from animal data, we can demonstrate that this durability appears to be lifelong. So we have a very large phase 1 study. It's actually somewhat of a phase 1/2 study because we have 2 components to this. One was a single ascending dose component and a dose expansion component. These were in both patients with polyneuropathy as well as in cardiomyopathy. In total, we've dosed 72 patients in this phase 1 study. I'll show you some of the data from about 60 or so patients from our phase 1 study, both from part 1 and part 2. We are looking at part 2 at a fixed dose of 55 or 80 milligrams, while we used a weight-based dosing in part 1, and we didn't find from a pharmacokinetic perspective that there was a difference, so we were able to move into a fixed dose, As most phase 1 studies, this is primarily a safety study, but key components that we were looking at were tolerability, pharmacokinetics, as I mentioned, as well as TTR reduction. We're also looking at clinical endpoints of interest, neurological endpoints, most predominantly the mNIS+7, as well as cardiac endpoints like BNP and troponin and 6-minute walk tests, and those will be presented at a later date this year. In terms of the safety profile, NTLA-2001 was generally well tolerated. What you see here is a table of treatment emergent adverse events by maximum grade. Many of these events are common for patients with ATTR, including cardiac failure and hospitalizations that were related to their disease. The most frequent adverse event is infusion-related reactions. This is a single dose, so this would only occur one time. The vast majority were mild to moderate, and no patient had any trouble completing their dose, so all patients completed dosing of NTLA-2001. As I mentioned, these were mild with no clinical sequelae. There were some elevations in liver enzymes that were observed. These were asymptomatic and required no intervention in order for treatment. So no steroids were related, and there were no hospitalizations. So patients had some transient increases in liver enzymes, which resolved on their own. This slide looks at the TTR reduction. And for those that are familiar with the currently available agents that are able to silence TTR, they typically look at the % reduction in TTR. But speaking again to a room of cardiologists, it's important that we think about TTR not in the % reduction, but also in the absolute level. So we wouldn't look at lipids in terms of what % reduction of lipids or blood pressure, the % reduction in blood pressure. We're very much interested in getting to the lowest level of TTR as possible because we believe strongly that that's going to lead to clinical benefit, and we see that in ATTR itself as well as other forms of amyloidosis. And so what we see at doses of NTLA-2001 of 0.7 mg per kg, 1, 55 milligrams, and 80 milligrams is that we achieve a very rapid reduction in TTR that's consistent across the patient population and durable. So what we see is an over 91% reduction in serum TTR, and the residual TTR that's remaining is about 17 micrograms per mil, which is the lowest that's been observed from any of the available silencing agents. I think the consistency in the reduction is also important. so that the vast majority of patients will derive benefit. And this was true across the cardiomyopathy patient population as well as the polyneuropathy patient population. So with the safety and the pharmacodynamic data, we're actually in a phase 3 study for cardiomyopathy currently. And so we'll have actually sites in Canada that will be participating as well. We've already launched the study, and it's actually enrolling quite well. Patients and investigators are very interested in putting their patients on NTLA-2001. It's a placebo-controlled, endpoint-driven study with randomization 2-to-1 at the 55 milligram dose, which we saw was both safe and established profound reductions in serum TTR. The primary endpoint looks at cardiovascular death as well as cardiovascular-related hospitalizations, such as heart failure, arrhythmias, which are common in patients with ATTR. Patients are eligible provided they have New York Heart Association class 1 to 3 and have NT-proBNP over 1,000, demonstrating that there's a continued need for these patients to have additional therapies. Some can be on background tafamidis if they're already on tafamidis. So patients in Canada that are on tafamidis would remain eligible for the study. It's an endpoint-driven study, so there's no fixed duration, but we expect the majority of patients to be on study for about 30 months. I'm going to move to NTLA-2002, which is our hereditary angioedema program. I don't know if this audience is familiar with hereditary angioedema. It's also a genetic disorder characterized by recurrence of severe and unpredictable swelling in various body parts, and particularly in the larynx. So that can be fatal for patients. It's a C1 esterase deficiency, which leads to increased bradykinin, which leads to the angioedema that you can actually see visually in many of these patients. There are available therapies, but there remains an unmet need. Patients continue to have breakthrough attacks, and this can lead to significant morbidity and mortality for this patient population as well. So we knock out the KLKB1 gene, which reduces kallikrein, which rebalances essentially the bradykinin to ensure that we reduce the incidence of attacks in these patients. It's also a single dose, so and we expect this to be lifelong for these patients, reducing significant burden and minimizing the risk of breakthrough attacks. We have an ongoing phase 1/2 study. Today I'll describe some of the results from our phase 1 study, and you could see that this is, again, a single dose therapy. We've dosed 25, 50, and 75 milligrams, so that's 10 patients. I'll show you the data from 10 patients, and we're currently looking at our phase 2 study data. The primary endpoint, as was with TTR, looks at safety, and pharmacokinetics and pharmacodynamic profile are also part of the key endpoints. But one thing that we can look at here is actually clinical outcome data as it relates to attacks. So that's swelling of body parts like the larynx or the abdomen or joints. NTLA-2002 was very well tolerated at all dose levels. Similar to NTLA-2001, the most common adverse event were infusion reactions. We had no adverse events that were above mild to moderate in severity, no SAEs. There were no liver enzyme elevations in this study that were above grade 1, so very mild liver elevations, and no changes in any coagulation parameters or platelets. This slide looks at the attack data, and so what we see is a 98% reduction in the monthly attack rate through the latest follow-up for these patients. So there are two ways to look at this data. One is at all attacks and attacks requiring on-demand treatment. So patients are really trained when they have either prodrome or they actually observe that there's going to be swelling, they give themselves rescue therapy. And you can see that for our phase 1 study, we were able to reduce both of those by 98 and 97%. So an extremely profound reduction in the attack rate compared to their baseline. This is a little bit of a busy slide, but I'll walk you through it. These are swim lanes. And what we see after that's 108 weeks, that 8 out of 10 patients are attack-free for the observation period. So it's quite a profound result. And how you read this figure here is to each patient is a swim lane. We can see the historical attack rates, some as high as 17 attacks. That's per month for these patients. And we have a screening period where patients are observed for a period of time, and you could see significant attacks for a number of these patients during that screening period. There's a primary observation period of 16 weeks, and then we continue to observe these patients following that primary observation period. You can see all the lack of color as you go further out, and that lack of color is indicative of the significant reduction in attack. There are a few patients that have mild attacks post that observation period. It's unclear whether those were true attacks, but they have not observed attacks in a significant period of time. Some patients are on prophylactic therapy as they enter into the clinical study, and all patients that were on prophylactic therapy were able to withdraw their prophylaxis and remain attack-free. So a really outstanding result, indicative of the profound reductions in kallikrein that we observe and the potential for a really profound benefit to patients. We're studying, as I mentioned, the data from our phase 2, and for both NTLA 2001, which is currently in phase 3, we'll also be advancing NTLA 2002 to a phase 3 program as well. The one part that I also want to mention as it relates to NTLA 2001 is that for the polyneuropathy indication, we'll also be advancing that hopefully this year into a phase 3 study. So with that, I'd like to, one, really thank the patients that participated. It's a really meaningful experience as a physician and someone in drug development to have patients that are willing to really volunteer and contribute to science. And so it's a real testament to the patient communities that we deal with and their clinicians as well, and of course their families. Thank you.
Thank you very much, Dr. Walsh.
Should I stand at the podium? Or?
Whatever you prefer. Yeah.
Yeah. I'm good either way. Yeah.
It's very, I mean, very impressive and exciting development. Looking forward to having drugs as developments for to target cardiomyocytes for the genetic heart diseases that we're interested in. I guess if there's any questions in the audience, please raise your hand. In the meantime, one question that's perhaps of general interest. What happens to the germ line cells? Do they change as well, and what's the consequences on the offspring?
Yeah, that's an important question. So regulations are pretty strict as it relates to editing of germline cells, and we've shown consistently across both NTLA 2001 and 2002 that there is no germline transmission, and we did that through non-clinical studies. And so from our perspective, and that's been agreed to by health authorities, that there is no risk of those therapies to edit germ cells. So there isn't a consequence then for our therapy and those patients.
just to put in perspective of what we just heard from you and the great work of your company, it's amazing. I mean, this is genetic Star Wars. This is patients with genetically predetermined inborn disease that can be cured or prevented from manifesting this genetically predetermined disease by a single treatment. Yeah. And it's just amazing that we're hearing this in our lifetime. I mean, the Human Genome Project that helped identify genes is only 30 years old. And so listening to you speak and present this is mind-boggling. I wanted to ask you, we welcome questions from the audience, but I didn't see anyone take a mic, so I'll ask this question. Along the lines of what Rafiq was getting at, the FDA will be reasonably nervous about gene editing, base editing therapies before approval for widespread use. Mm-hmm. The offspring or the off-target issue is one thing. What else do you see as potential barriers or issues that you have to satisfy the FDA for before this becomes widespread?
Yeah, I mean, that's a great question and a really important one. Can you hear me? Yeah, that's better. I think there are a number of different components that relates to clinical, non-clinical, in addition to CMC, which often sort of gets overlooked. So the manufacturing components. The FDA is going to want to understand that this is a favorable benefit risk. I mean, that's sort of clear. From the non-clinical perspective, ensuring that there is a very low risk of off-target potential, germline transmission, is going to be really important as well. For all gene therapies and for gene editing as well, clinical studies, even after they've been approved, need to be conducted in order to follow patients for up to 15 years to ensure that there are no unexpected safety risks. They'll approve the therapy, but they want you to continue to follow patients. I think that's to our benefit as a community, also to Intellia. We'll understand not just long-term safety, but also long-term efficacy. Most clinical studies aren't following patients out sort of that long. So I think that's a real important consideration. As it relates to manufacturing, one thing that the FDA is very clear on is the consistency of your manufacturing process. Is what I used in our phase 3 study now going to be consistent, comparable to what we're doing in the commercial space when this is actually being used in the real world? And as my first mentor in industry told me, the biggest study actually is when the drug is released into the real world. And so one part is related to the manufacturing process. We've done an excellent job of ensuring that we have consistency between batch to batch. We can generate data that supports that. You can understand why the FDA might be concerned, and other health authorities as well. Because of the work that we've done to date, even for getting into phase 3, this has been something that we've worked very hard to ensure, for patients' sake, frankly, that we are providing them the best therapy that's consistent from batch to batch. So I think that's part that's often overlooked and probably doesn't get enough attention.
I've got a quick, so incredible, and I think it's incredible that we're entering this era. With CRISPR nucleases, you get a double-stranded DNA cut and then non-homologous end joining to repair it. In terms of the indel that you generally get, I remember for the amyloid paper, it was kind of one general frame shift in almost all of them. Is that what you guys are typically seeing after it? It's just one? Because I guess people talk about you could have any number of insertion or deletions, but is it generally one product? And after that as well, just in terms of commenting on, with the double-stranded DNA cut, the possibility of getting translocations that people have worried about, you guys, I guess, are not seeing any of that?
Yeah, that's correct. I'll answer the translocations first. We don't see that across multiple sort of different in vitro studies that we've done, across different cell lines, across different donors. from different tissue. And we don't see sort of differences in from the New England paper that was recently, I don't know if you're talking about you mentioned the ATTR paper. It's the same as it relates to the HAE paper as well.
The not homologous end joints, not that chaotic.
This is, it seems to be that selection of the guide is really important, but I think the concerns that you might be hearing just don't play out within the work, at least that we've done.
We still have time, Jason?
Oh, yeah. Go ahead.
Anybody from the audience? I know you're here mainly to talk about the science, and you did a great job doing that. But I want to bring us back to the economics a little bit, and I apologize if you're not prepared to discuss the economics of this type of therapy, but it's of course very important, particularly for the hereditary TTR group, which is a population of patients that demographically are a little bit more impoverished. Do you guys give thought about the economic feasibility of this for the patients who will need the therapies? And I know price ranges are not something you're probably thinking about, but what are your thoughts on this?
So it's an important question. We do think about what it means to deliver value to patients, to the healthcare system in general. And so I won't get into necessarily how we price this, but we're aiming to bring this into the market in order to treat the maximum number of patients possible. So demonstrating value comes with demonstrating strong efficacy, that the profile is safe, and for a gene editing therapy, or even any gene therapy, we really have to demonstrate the durability of the therapeutic as well. I think as it relates to the sort of underserved populations, that's a question that I think all of us here will have to sort of work together in order to address how to deliver this therapy in an equitable way. Certainly at Intellia, we're thinking about how do we do that. And I don't know if we have the answers yet, but it's an important part of what it is that we do.